Okay, so good morning, everybody, and welcome to the second day of the MSG. I'm really pleased to welcome you all to what's going to be a really exciting day. As you can see, I'm very briefly in a car, and I apologize for that. But we are going to have a few introductory words from me and from Rick. First of all, just to update you and to welcome all the international fellows who are part of this new international center that was established about a year ago, funded by the Medical Research Council. I just have a couple of slides to tell MSG members about that and how they might become involved, as well as to welcome international fellows appointed onto this program who I know are dialing into this meeting. Next slide. This centre kicked off a year ago, funded by the Medical Research Council in the UK. Next slide. And essentially it's to build a fellowship program between the countries involved and to develop and train fellows in neuromuscular genomic medicine. The fellows based in each of their countries will build cohorts of children and adults with neuromuscular disease and deeply phenotype them, and also undertake linkage into a genetics program in the UK and in some of the host countries. And we've established a bioinformatics platform and a genetic program. And we hope this will help us develop a better understanding of genetic architecture of these diseases, as well as enable diagnostics in these different countries and build trial-ready cohorts. Next slide. So colleagues from all of these countries I know are dialing in, and they're very welcome to the MSG. So colleagues from Brazil, from India, next slide. Colleagues from South Africa and Zambia, as well as Turkey, and colleagues in the USA, all illustrated here. Next slide. And it links with 3 centres in the UK, Newcastle, Cambridge, and also UCL, where I'm based. Next slide. So it's really just to make MSG colleagues aware of this new initiative, and particularly to welcome all of the fellows who are dialing in, and we hope we'll get a lot of enjoyment and education from this MSG meeting. There are 12 international fellows, as well as their PIs in each country. And if people want more information about this, Lindsay Wilson is the person to contact. Okay, so I'm going to hand over to Rick for a few words from him. Rick.
All right, thank you, Michael. Well, good morning and good afternoon, MSG. Michael, there was a really great picture of you in that last slide. It looked, you really cleaned up in that picture, the car picture. So it's great to have everyone here on day 2. I think yesterday was phenomenal, and we're so excited to have everyone sign in for today's event as well. I had just a very couple slides like Michael, sort of informational slides. Can you give me the first one, Gil?
First one's up.
And the next.
Oh, that's it?
Oh,
okay.
yeah.
Yeah, that one. So
Okay.
this is, so Michael has these very fancy international organizations that he leads. Mine are a little more lowbrow. So this is the RNNMF website, which most of you know about, but there may be a few that don't. And so this is how to get on that conversation, to talk about cases and to see grand rounds and everything, rnmf.com. And there's also, they're less active, but there's an autonomic, an MS, an epilepsy, and a headache site too. And then the other, next slide is once again to plug our new journal and the RNMF Neuromuscular Journal. And so we have issue 4 came out this week, and it's the MSG issue with all of your abstracts. And so we're really excited about that. All right. So, and then finally, you know, this is probably not relevant to most of you experts that are signing in for this MSG annual meeting, but your registrars, residents, fellows, medical students, make sure that they know about our neuromuscular review course, which is, they take place on Saturdays. They're about, they're they're divided in two parts. They're each about 3 hours. And they're great introductions to the concept of neuromuscular disease for the clinician. Directed by Mazen Damachki, I'm involved, John Katz, Todd Levine, and they're $25, but we waive it for trainees. So this slide we'll put on the website too, so your trainees can know how to sign up for these courses. There's 4 more this year. All right, so that's all the informational stuff about other neuromuscular things going on. And now it's a great pleasure to introduce this year's Robert C. Griggs, MD, annual MSG lectureship. And before I introduce our speaker, let me just say a couple words about the lectureship. So we have Dr. Griggs on the call, and he's got his camera on. So wave, Dr. Griggs. Thank you. And it's for most of you know, but there's a few of you, this could be your first MSG meeting. And Dr. Griggs is the founder of the MSG. He got Dr. Hannah and I and Dr. Twill, our speaker, and many of us involved in neuromuscular disease early in our careers. And he started this organization in the, I think it was 99. Although we probably need a historian for the MSG, Dr. Griggs. I'm not sure that we've ever actually written the year by year initial history of the MSG. But it started in 99, I believe, with a meeting in Rochester, and then we quickly moved to Beaver Hollow in upstate New York, and Dr. Griggs led the meeting there for about 15 years. And when he stepped down as the chair of the MSG, and Dr. Hannah and I took over as chair and co-chair, one of the things the MSG leadership decided to do was to honor him by creating this Robert C. Griggs, MD, annual MSG lecture. So we should have had a slide. Actually, next year we'll do this. We usually do it when we have it in person, of the former Griggs lectures, but they're all extremely prominent leaders in the field of neuromuscular disease. And so this is the 4th one. So in that vein, it's just an honor to have Dr. Robbie Twill as this year's annual Robert C. Griggs MSG lecturer. Dr. Twill is professor of neurology at University of Rochester, and he's the Fields Endowed Professor of Neurology. Actually, Robbie, I don't have your CV in front of me, so I'm going to wing this a little bit, just because I've known you for over, well, probably close to 30 years. And Robbie did most of his training in Rochester and was one of Dr. Griggs's mentees and proteges early on in neuromuscular disease. And then Dr. Twill began working in the field of FSH early in his career and began collaborating with other FSH researchers around the world, and particularly in Europe. And over the last 25 years, he has become probably the world's leader, or one of the a handful of world's leaders in the field of FSH dystrophy. He's trained legions of fellows who are in the field of FSHD, and it's totally appropriate to have Dr. Twill not only as this year's Griggs lecturer, but also as probably the world's leader in knowing about FSH, so that he can give us an update on that to the MSG this year. So Dr. Twill, I'm going to turn it over to you, and thank you for doing this, and congratulations.
Thank you, Rick. Should I?
Yeah, you can pull up your slides now.
Yep. You have my slides?
Do you want me to pull it up for you?
No, I mean, I'm
We don't see it yet.
okay. Here we go. Do you see it?
No, did you share screen yet in Zoom?
Yeah, I did.
Oh, we don't see it yet.
Once when this happened yesterday, I think one of the speakers, we actually ended up having the speaker email the slides to Gil.
I have his backup in case, but he should just see, I mean, there should be just a menu prompt somewhere to share the slides. So,
Yeah, I did that, but it's
Well, if you have a backup, Gil.
yep. All right. 1 second.
Gil, I did send Liz an updated.
Yep, I have the latest version. It's coming
Okay,
right.
great.
It's coming up right now.
Awesome.
Okay. Well, thank you, Rick, for this introduction. I was surprised when you first asked me to do this. I didn't realize I was old enough to be doing this until I realized that I've been doing this for 29 years. It's a special honor for me in particular because Dr. Griggs was my mentor for many years early on in my career. And I give him a lot of credit for where I am right now. So, Gil, can I control the slides or?
You'll have to prompt me.
Okay, okay, go, yeah, okay. What I would like to do is give you a brief summary of the clinical manifestations and disease mechanism of FSH to start off, and then talk to you about how we got here. FSH research from 1990 to 2020. FSHD has been a particular difficult nut to crack as far as trying to figure out the disease mechanism. And so it required a lot of patience and perseverance. As a clinical researcher, you can't really walk away from a patient population that you've worked with for many years. So even though things did not, you saw no light at the end of the tunnel at some point, you had to persevere and keep going. But one of the things I want to relay from this presentation as well is really the joy and reward of collaborative research. And I put an asterisk there because you really need to choose your collaborators. They have to be people who you can work with, who are transparent, and you have to be all on an equal footing when it comes to the work that you do together. Next. So FSHD is one of the most common adult forms of muscular dystrophy with a prevalence of anywhere between 1 in 8,000 to 1 in 20,000. That the 1 in 8,000 is a more recent study done in the Netherlands, and probably the most accurate estimate to date. The inheritance pattern, as most of you know, there are 2 forms of FSHD. 1 and 2, the FSHD accounts for about 95% of all patients with this condition. It's autosomal dominant, and FSHD2 has a digenic inheritance. Next. So the clinical features, there's a distinctive pattern of weakness at onset. Face and shoulders are involved first with a characteristic shoulder profile. It progresses to involve the muscles of the trunk and the lower extremities. A wide spectrum of severity from minimally symptomatic to wheelchair bound, and about 20% of patients above the age of 50 lose the ability to walk. There are interesting non-skeletal muscle involvement, and the 3 main ones are the hearing loss, retinal vascular disease with an exudative retinopathy called Coats disease, and also cognitive impairment. All those three non-muscle involvement are restricted to the patients who have the most severe disease. And even in that category, there are very few who have this combination of symptoms. And I'll get back to that at some point. So just a few images of typical patients with FSH. On the upper left-hand side, you can see a patient with FSHD, and I'm asking her to close her eyes. You can see that she has weakness of the upper of her eye closure, but as she's wincing, also you can see an asymmetry in her in the movements of her lower face as she's trying to whistle. Below, also you can see that she is her lips are weak as well. Below that, you see a very characteristic profile of the shoulders viewed from the front, where you see straight clavicles. You can see also atrophy of the pectoral muscles. In the middle, you see the limitation of shoulder abduction because of the scapular winging, and you can see a prominent scapula behind him. And you can see again the asymmetry in when you look at the backside of that same person. One scapula is winged and the other is not. And again, some of the more severe aspects of the disease as the development of a fairly significant lumbar lordosis. In the lower right side of the slide, you will see identical twins, and all I've asked them to do is flex their arms. And you can see that on one side, they are less than anti-gravity in their biceps, and in the good biceps, it's 5/5, so full strength. And again, these are not only interesting clinically, but I think there are some implications as to how the disease mechanism operates. Next. So getting to the bottom line, the disease mechanism is an inappropriate expression of the DUX4 gene in skeletal muscle. DUX4 is a transcription factor that plays a role in embryonic development normally, and it's only expressed in the testes and thymus, but not in other somatic cells. It is also notable that the DUX4 protein, the DUX4 gene, is present in humans and non-human primates, but not in other animals. And this is important because that's one of the challenges in FSH research, is that there's no mouse model or any other animal models that really reproduces what happens in FSHD because of the absence of that gene in this in mice and other animals. So the inappropriate expression of DUX4 in skeletal muscle activates a number of genetic programs leading to inflammation, muscle atrophy, increased susceptibility to oxidative stress, and disrupted myogenesis. So the gene is located on the tip of the long arm of chromosome 4 at 4q35. There are multiple tandem repeat copies of DUX4 contained. Each copy is contained in a 3.3 kilobase macrosatellite repeat each unit is called a D4Z4 repeat. And at the tip of chromosome 4, Q35, there are between 11 and 150 repeats in normal individuals. And when you cut that down to 1 to 10 repeats with the appropriate background, you get FSHD. Next. So here's an explanation of the genetic mechanism of FSHD. And again, what you're looking at is a chromatin structure, a cartoon of that with the D4Z4 repeats as red triangles initially. And again, the repeats occur with two potential polymorphisms at the distal end of the repeats of 4QA or 4QB. The 4QA has a polyadenylation sequence, whereas the 4QB does not. And the polyadenylation sequence is important to stabilize our mRNA and allow the production of a full DUX4 protein. On the right side, you see what happens with a 4QB, contraction of the repeat. You open up the chromatin structure. The mRNA is produced, but it's unstable, so you cannot produce a DUX4 protein. Whereas if you have the 4QA polymorphism at the end of the repeats, as you see on the left side, with the contraction of the repeat, you open up the chromatin structure, make it permissive for transcription translation, and so you produce an mRNA which is stable, and therefore you can produce toxic DUX4 protein. Next. And FSHD2 is different, but it really kind of supports the disease mechanism of FSHD1. And in FSHD2, you have a normal number of repeats. And if you go to the right side of this, to this figure, you see a normal number of V4Z4 repeats. The blue oval-shaped structures on top of that are this SMCHD protein. SMCHD gene mutation is associated with FSHD. Most patients with FSHD2 have a mutation in that gene. That gene is important in DNA methylation and exon activation. So it's important in keeping the chromatin structure hypermethylated and therefore closed up. And so anything within that structure is not able to be expressed. On the right side, what you see is if you have a mutation in the SMCHD1 gene, you open up the chromatin structure here. And again, if you don't have a permissive poly-A polymorphism at the end, you produce mRNA, but no DUX4 protein. On the opposite side, if you have a polyadenylation sequence polymorphism at the end, then you can produce a toxic DUX4 protein. Next. So again, both in FSHD1 and FSHD2, both, even though there are distinct mechanisms, the end product is the same, and it leads to the expression or the inappropriate expression of DUX4 proteins in somatic cells. Next. So the linkage of FSHD to chromosome 4 happened in 1990, and I started my fellowship in 1991 with Dr. Griggs. Under Dr. Griggs' mentorship, I started working with folks at Ohio State University, was John Kissel and Jerry Mandel, to identify and characterize clinical features and genetics of as many families as possible. We had collected up to more than 100 families at that point. We tried to basically clinically characterize those families as well as do genetic testing on all of them. And this required often travel on my part, and one of the most interesting travel that I did for tracking down a family from Pennsylvania that had extended families in Arkansas and in Nebraska. And so I went to Arkansas and then to Oxford, Nebraska, to family gatherings, examined everybody and collected blood samples. And my return to Omaha from Oxford, Nebraska, I had to get one more person from that family who was actually a pastor in a church who was giving Sunday Mass. So I had to wait for him in his office, examine him, and take a blood sample. And you can imagine what would happen now if I had to go through airport security with a bag full of syringes and dozens and dozens of tubes with blood. So that was very productive, and we collected many families at that point. The one reference I had at the start of my fellowship is a photocopied copy of Dr. George Padberg's PhD thesis that really this is the best clinical description of FSHD that anybody has done. And this was always my reference point, and everything that he had in there, almost everything, turned out to be exactly right about FSHD, its clinical manifestations. And so this is available online for anybody who's interested, and it's a really valuable resource. Next. So in 1993, after localization to chromosome 4q35, there was the actual genetic lesion was identified in 1993, and was a contraction of a macrosatellite repeat chromosome 4q35 as the cause of FSHD. This development made the conduct of a natural history study in FSHD all the more urgent for us, because at that time we, as many people thought, that that the identification of that actual gene was just around the corner. Next. So we did a joint natural history study at URMC and OSU, recruited 80 patients, were followed for up to 3 years. And we could demonstrate that there was a slight decline in strength on manual muscle testing QMT over 12 months. This actually is the one and only natural history that has been done since then until more recently, and we've been involved in a recent natural history study that I'll be talking about a little bit later. Next. One of the things that had been left unanswered is the use of prednisone in FSHD. This came from the fact that inflammatory infiltrates in FSHD are quite common, and in the 1970s and 80s, there are many case reports and case series showing that these patients are responsive to prednisone. So we did an open label study and basically showed that there was no benefit to prednisone except that making people gain weight. And so we put that aside. And again, as we were waiting for an identification of the disease mechanism, we were thinking about a possible intervention. Next. So we did, John Kissel and I were thinking about what type of intervention we would be interested in. And so we were actually interested in using clenbuterol, a beta agonist that is known to have anabolic properties, but we couldn't get it at that time as it was not available in the US. So we used albuterol instead and showed slight dose-dependent increase in muscle mass and a modest improvement in grip strength. Next. Next as well. And as you can see here, we could show a definitive statistically significant increase in body mass, lean body mass at the end of 1 year. This is particularly interesting now because we know that the beta agonists are actually not only anabolic agents, but in relation to FSHD in particular, drug screenings have shown that albuterol and clenbuterol both not only can actually block the production of DUX4. The albuterol study was replicated in the Netherlands and showed similar results. And another study was done in France, but the one in France actually did not show a significant change, but there were a lot of problems with data collection in that study. Next. So between 1993 and 2002, there was no clear disease mechanism. The DUX4 sequence contained within each repeat lacked some components of what we would consider an expressed gene. Next. No one could reliably measure DUX4 in cells or tissue. It was proposed then that the loss of the critical number of repeats could either facilitate or repress expression in genes proximal to the DUX4 repeats. Then this case report was published in 1996 by Rosella Tupler and colleagues, and they showed that a patient who had monosomy of, deletion of the distal 4q did not have FSH muscular dystrophy. And that again suggests, as we know now, you need at least one D4Z4 repeat to cause FSHD. This basically told us that you need the DUX4 repeats to cause FSHD, and because so it's a deleterious gain of function from the DUX4 at that point. That would be the conclusion from this study. Next. So what we did from 1996 to 2002 is we examined the genotype-phenotype relationship between in patient populations that we had and determined that there was a correlation between the residual repeat size and the disease severity. Next. Next. And one more. And you can see on the right side, you can see the distribution of age at first wheelchair use, and you can see that patients who have 1 to 3 repeats are likely to get become wheelchair bound at an earlier age, more than 4 to 7 repeats, and the mildest patients are those who have 8 to 10 repeats. You can do the same with age of onset, and you see the same distribution, or disease severity, you will see again the same distribution. And the patients between who have 8 to 10 repeats are also more likely to have non-penetrant disease. And typically also women tend to manifest the disease later than men, where men manifest in the late teens to early 20s. In women, it's about a decade longer. So in the late 20s to early 30s. As research in FSHD molecular genetics stalled, we thought that it was essential to collect biological samples. And so one of the things I focused on, and again, this came again from the realization that there was not going to be a good animal model for FSHD, so the human muscle samples were going to be very useful. Also, we had a lab that had technicians who were very skillful at creating myoblast cell lines, so we collected a large number of samples, over 150 muscle biopsy samples, from which we created myoblast cell lines, flash frozen samples, and samples for histology. And these have proven to be very useful in trying to figure out the mechanism of FSHD, and more recently in drug screening, and has become, the myoblast cell lines have become very popular among the drug companies that are interested in entering the FSHD treatment. We also showed that FSHD myoblasts are more susceptible to oxidative stress. That was also documented by other researchers since then. And with the help of Dr. Charles Thornton's lab, we performed one of the early microarray expression profiles in FSHD. Next. In 2002, there was a proposed disease mechanism. This is published by Gabellini et al. And the mechanism, the proposed mechanism is that the genes proximal to the D4Z4 repeats are activated by loss of the D4Z4 repeats. The presence of the D4Z4 repeats inhibits, by this theory, is that the DUX4 repeats, they're heterochromatic DNA, and the proximity of the genes to the D4Z4 repeats actually are inhibited by the D4Z4 repeats. And what happens with the contraction of the repeat is that you allow the production of some of the, or the increased production of some of the DUX4, the genes proximal to the D4Z4 repeats. One of the major problems is what we mentioned before, is that deletion of all of the repeats on chromosome 4 does not lead to FSHD. But Gabellini made the argument that that person reported to have a deletion of D4Z4 repeats also had deletions of the FRG1 and FRG2 genes that are more proximal. And so the idea was that FRG1 gene overexpression is what caused FSH muscular dystrophy. And there was a mouse model of FRG1 overexpression that showed a myopathic profile. However, the FRG1 expression in the mouse was multiple times higher than than what we saw in human muscle. Subsequent studies have supported or disproved that this disease mechanism. There was a lot of controversy about this. One of the issues that this was kind of unfortunately, the working disease mechanism for the next decade, and hindered actually the funding of alternative hypotheses for the disease mechanism and FSHD. In 2003, Richard Lemmers from Dr. Vandermarle's Leiden lab described a patient who had a proximal mutation to the D4Z4 repeats that included all three of those genes, FRG1, FRG2, and AT1, and showed that they had a contraction, who had a contraction of the D4Z4 repeats, and they had FSHD, again, inferring that the FRG1 was not needed to produce FSH muscular dystrophy. Next. So for our research, the turning point really happened at following a small meeting set up by a family foundation in Seattle, where Sylvain Vandermarle was present, who's a molecular biologist at Leiden University. And in discussions with him, we decided to collaborate. Sylvain, as some of you know, is a superb molecular biologist, but he's also actually very in tune to the need for biological samples from humans, and the importance of characterizing as many patients as possible and looking for patients with unusual molecular pathologic changes. Next. So in 2004, the Leiden lab published a paper showing that only contractions in D4Z4 repeats with an A variant, as I showed before, though that's the permissive variant that has a polyadenylation sequence after the last repeat, can cause FSHD. Next. In 2007, Richard Fields, who's a very wealthy real estate developer in New York City, approached me with an interest in funding a center for FSHD research in FSH in Rochester. And I proposed creating a center without walls that included Leiden with Sylvain Vandermarle, and Seattle with Stephen Tapscott, who's a prominent cell biologist that I started working with as well. This came my institution was not very happy with that, because as we got money from Richard Fields, the first thing I planned to do is give some of it away. But that was the right thing to do, because trying to set up a basic scientist at Rochester would have taken a lot of money and many years of catch up for that person to reach the point where Sylvain Vandermarle was at that point. In 2009, in collaboration with Leiden, we looked at expression in the genes proximal to the repeats in myoblasts, myotubes, and muscle. And we did that both at the RNA and protein level. And we could show, with the exception of FRG2, none of the other genes proximal to the D4Z4 repeats showed any consistent increase in expression, and we use controls as well as myotonic dystrophy samples as well. FRG2, however, was increased, and later on we found out that FRG2 actually is one of the target genes for LUX4. In 2010, the Tapscott lab published a manuscript showing expression of LUX4 in myoblast cell lines and in myotubes. Next. And one of the difficulty that plagued the research in FSHD is that even people who are able to measure LUX4 levels, the LUX4 level was extremely low, even at the RNA level. But what Stephen had done is taken myoblast cell lines and picked samples from different parts of the cell lines, and he was able to show that in only a minority of patients do you have LUX4 expression. You can see that on the left side, and many of the cells showed no expression. So this is called jackpot PCR, and basically shows that only minority of cell lines, of samples within those myoblast cell lines showed a robust expression of LUX4. You could also show on the right side, if you let the myoblasts evolve into myotubes, that you can show very clearly expression of LUX4 in the pink in the nucleus of the myotube. Next. In 2010, a unifying FSHD genetic hypothesis was published. This is based on the research, again, done by Sylvain and all of his collaborators. And the way this was proven and beyond any doubt is that Richard Lemmers was able to show that in patients who have a translocation of the D4Z4 repeats from chromosome 4 to chromosome 10, if they had the translocation was of a contracted D4Z4 repeat with an A allele, then you had FSHD, meaning that the adjacent genes on chromosome 4 played no role in the production of FSHD. This led to a lot of headlines in the news about a reanimated junk DNA causing disease, or the zombie DNA causing muscular dystrophy. There was still some resistance to this mechanism. Next. And next, yeah. Yeah, one more, I think. Okay, so in 2012, again from Sylvain's lab, they published, they were able to show that SMCHD1 gene is the major cause of FSHD type 2. Again, 95% of patients with FSHD2 have this mutation. And the finding, again, further confirms that the end mechanism of both FSHD1 and 2 is the activation of the LUX4 gene. that is not expressed in somatic tissue. Since then, there were 2 additional FSHD2 genes found, DNNTB3 and LRIF1. Both of those, again, just like SMCHD1, are important in a chromatin structure and methylation. Moreover, it was shown that there are rare families who happen to have both FSHD2 and FSHD1, and that SMCHD1 can be a modifier of FSHD2, and you can see in this family tree patients who, so a person with FSHD1 with a spouse who has FSHD2, and their offspring who have 1 and 2 have much more severe disease than patients who have one or the other. Again, this opens the field, to the fact that there are probably many other modifiers of disease severity in FSHD, and that might explain the intra and familial variability and even the variability within patients with the same number of residual repeats. Next. So at that point, in my pursuit of FSH muscular dystrophy, this 2010 became the time when, as a clinical researcher, I became very excited and to kind of dive right in. Jeff Statland at that point joined me as a neuromuscular fellow, and within 2 weeks started working on plans for FSHD trial readiness. We had, and again, as many of you know who've tried to get such grants, there's a lot of resistance in NIH funding. There's no particular program to study to get trial readiness for a particular, to achieve trial readiness for any particular disease. So we settled on foundation money, allowing us to do small studies. And one of the things that we thought was very important is to start a nidus of a clinical trial network of 4 sites. And we did that with private funding. And then next, in 2015, NINDS and NCATS announced a clinical trial readiness UO1 for a rare disease program again, and we applied for that and got in the first and only UO1. We got the first and only UO1 in that first funding cycle. This led us to expand our clinical network to 8 sites in the US and 3 sites in Europe that were involved in our natural history study. We now have 12 sites, and we're going to add soon another 3 sites in Europe, and we're going to have a robust number of sites, all trained similarly to go forward with clinical trials. Next. One of the things that, again, coming back to DUX4 is difficult to measure in muscle and even in cell lines. And so one of the things that was important is development of tissue biomarkers. Stephen Tapscott taking some of our muscle biopsies from controls and FSHD, as you can see on the right, determined that DUX4 being a transcription factor turns on many programs. And if you can't measure DUX4 in a quantitative way, he suggested that there are 4 selected biomarkers, all of them targets of DUX4 that can be measured successfully. Again, please, yep. One of the things that you can see on the bottom of this graph is that the C is a control patients. These are normal individuals. But there are patients with FSHD who overlap with, as far as their profile, with normal individuals. This kind of raised the issue of the fact that there are some, the profile in some muscles in patients who have FSH looks like normal, a normal control. And again, subsequent to that, next, an Italian group who was working on MRI did muscle biopsies of stir-positive MRIs and showed that stir-positive MRIs reliably showed, actually, I don't remember the number of samples that they did, but a small number of samples showed that all of them had that DUX4 target expression that Stephen Tapscott had shown. So we set up to kind of expand on that and try to develop a reliable biomarker. And what we did is do MRIs of the lower extremity muscles, choose muscles whenever we can that are SIR-positive that we can access with a needle biopsy, do RNA-seq pathology, and then repeat that in a year. And what we were able to show is that indeed there was a correlation between pathology, MRI, STIR positivity, and the reliability of our ability to pick up the DUX4 target expression. Next. And so again, so this biomarker is still in the process of being kind of perfected, and we're doing further studies with that in a second iteration of our Wellstone study with Seattle. So closing the circle, there are some things that are still of importance and that we have not answered, I think. So how does stochastic expression of LUX4 myoblasts and variable expression of LUX4 targets in muscle inform us of what we see clinically? And so what does it tell us about a couple of patients here that I've seen? The young high school volleyball players who presented with a unilateral foot drop that occurred over weeks by her report. But on examination, she couldn't lift her arms beyond 90 degrees. She played volleyball, I have to remind you, but had no concept that she had that limitation. And again, this suggests that there is a stochastic expression in particular muscles that leads to both asymmetry and also this the sense of muscles turning on and turning off DUX4 expression, and showing this variability in the progression, even in patients who start with, as is the normal in involvement of the upper extremities, and then you see them 20 years later, they may not have significant worsening of the upper extremities, but they have severe weakness of the legs. Again, another example of an extreme patient is a patient I saw at the age of 3 years, had one residual repeat. He had hearing loss, Coats disease, and one was cognitively impaired. I followed him for another, until he was 11. At that point, I was expecting that he would be very significantly involved as far as his muscle weakness. He had absolutely no muscle weakness except for some facial weakness. Again, these are issues that we need still to resolve. There are certainly other factors that are affecting the way disease progresses in FSHD and how individual muscles are involved. Next. So I want to acknowledge my collaborators, Sylvain Vandermal and Tapscott, Stephen Tapscott in Seattle, but also now joining me for the last 10 years, invaluable input from Jeff Statland, but also from all of the other folks at the University of Rochester, from Mike McDermott, who's a biostatistician who has helped us many times when we didn't help in our projects with FSHD, and Katie Eichinger, who was a physical therapist, an outstanding physical therapist who's training all of our centers right now, and again, Leanne Lewis and the other lab folks at our institution who have done a superb job with the myoblast cell lines. And again, this could not have been done by the commitment of hundreds of patients who have contributed to our research, and where without much prompting, where many patients have given us multiple pieces of their muscle that have been very valuable in getting to this point in our research on FSH muscular dystrophy. Thanks. Next. Do I have time for questions?
Thanks, Robbie. That was terrific. Questions for Dr. Twill? There's some Q&A. Let's see. Tom Lloyd writes, Robbie, what are thought to be the critical downstream targets of DUX4 that mediate toxicity?
Yeah, so I mean, that's an important question. I don't think we know that. I mean, there are so many genes that are turned on by DUX4. It's hard to know which one is the culprit or the one that is responsible for most of the pathology in FSHD. That's why it's, you know, targeting more proximally at the production of DUX4 is really going to be the way to go. Second is from Jeff Rosenfeld, any insights as to why the facial weakness and fibular weakness are so variable in the phenotype? Short answer is no. I don't know. And again, I think we still have to answer that question. I don't think we have an answer to that. And it's still, I think you see a lot of variability in many muscular dystrophies, but the changes within individuals and within groups of patients with FSH is really, I think, very specific for FSH muscular dystrophy. And again, it has to do with this stochastic expression of DUX4, I think. There's something that turns on and turns off DUX4 in muscles at different time points.
Yeah, when I give my basic neuromuscular talk, after I go through all the phenotypes, I said someone's going to ask why. Why is some face? Why does IBM have a phenotype? Why did the dystomorphy have a phenotype?
So I have a question, Rick.
Yeah. Go ahead.
So Rabi, your talk is terrific, and it's really a lesson for young people. The number of times that you were elated because it looked like we found the answer, and then turned out we didn't, but never gave up, and the number of times you were slapped down by various granting mechanisms and other things, but never gave up and continued to move forward is a real lesson to people. Never give up. But the question I have is, do you think that DUX4 explains everything, that all mechanisms that change the phenotype in terms of its distribution or its the other things that go associated are all mediated through DUX4? Do you think it's possible that there are other genes? Because in most other diseases, we've found that there are other loci, other genes that are important. And I've wondered, one of the things you didn't say much about, but which patients will tell you, and I'm sure you've heard this too, is the paroxysmal expression, or the paroxysmal worsening of the disease in an individual muscle, or the course accelerates. And it almost makes one think of the channelopathies, where the gene is necessary but not sufficient for an attack. Here I wonder if DUX4 is necessary but not sufficient for some of the manifestations we see, and there might be other genetic mechanisms or even environmental mechanisms that alter the course of the disease.
Yeah, absolutely. I mean, that's why I gave that example of this young woman, a high school student who came with a foot drop. And again, I think everybody who's seen patients with FSH will have that, patient, they'll hear it from the patients that they have sudden worsening. And yes, I think there are other factors, and there's something that affects, that triggers the turning on of the DUX4 gene that causes the damage. I don't know what these triggers are and why are they so specific to a particular muscle at any one time. And I think these are all unanswered questions. So again, this comes to Hani Kuslov's question. You're looking for additional genes that cause FSHD. I think that has been done before. I mean, there's not that has been done in the past, and there has not been any potential link to any other gene at this point, other than those associated with FSHD2. And the last question was from, do I recommend albuterol for all my FSH patients? At this point, no. I mean, I think we did a study. They did not, it was not beneficial at the end of 1 year. It is though, I mean, it is kind of intriguing that you can get to that point. But again, one of the issues when we saw that there was that kind of a flattening of the curve after 6 months of improvement, that maybe you downregulate the beta receptors if that was the mechanism, and that you don't get any more benefit after that. However, again, I mean, we are interested again in looking at that. Clenbuterol is a much more powerful anabolic agent, also shows in drug screening that it's a powerful inhibitor of DUX4 expression. So that's something that we may pursue.
Any other questions? Oh, we got Dr. Twill and Dr. Griggs here. Birch, I do believe that Robbie is the first annual Griggs MSG lecturer who was one of your trainees. So this is, I think that means we're getting old.
You're gonna give
But
him
it's also a credit to you, Dr. Griggs.
Give him his award, Dr. Barron.
Yes, so here is your virtual award. It's in the mail.
Thank you.
And congratulations again for amazing work in this field, and for bringing so many along with you. Thank you for training Dr. Stoutland.
Thank you for sending him to us.
Yes. All right. Actually, we're a few minutes ahead of schedule, and I think we wanted to maybe stay on time. Here, Lindsay, you're the moderator for the next session, Lindsay. What do you think we should do?
Yeah, I think we might as well move along, and then worst case, we can break early.
Okay.
Great. So with that, we'll transition into the next part of our agenda.
Hey, Lindsay, can you introduce yourself to everyone? Because we have so many people on. Yeah, thank you.
Absolutely. So I'm Lindsay Alfano. I'm a physical therapist at Nationwide Children's Hospital in Columbus, Ohio. Been part of the MSG Executive Committee and now have had the honor of being part of the Planning Committee. So with that, I'm really excited to move into this next section of our agenda. I think we've got some great presentations planned, and hopefully we have some continued discussion as we've had already this morning. So remember, you can put some questions in the Q&A section. You can also raise your hand at the end of the presentations if you'd like to ask your question via the audio features. So our first presenter is Karen Lee from the Canadian Agency for Drugs and Technology in Health. And she's going to be talking to us about rare diseases and why consider economics.
Karen, you can pull up your presentation.
Great. Great. So hopefully you can see my slides. Yeah.
Perfect.
Great. Thanks. So I was asked to speak to you today about rare diseases and health economics. And so this is probably a bit of a change of pace from the previous presentations you've had so far for this meeting. But there's often a lot of questions that we receive, especially at CADIF, regarding why even consider cost effectiveness or economics pertaining to rare diseases. They're not going to be cost effective. What information would we actually glean from doing this? And so I'm going to put it out there that it's still valuable, especially when we're thinking about making decisions about very costly interventions, that we should consider it. And hopefully through this presentation, I'll explain why. Just by way of disclosure, I don't have any conflicts to my knowledge, and I didn't receive any funding for this presentation. So as mentioned, I work at CADIS, which is a Canadian agency that provides Canadian healthcare decision makers with information, both clinical, economic, on which to make decisions pertaining to the optimal use of drugs as well as medical devices. So if you take a look at our website, we do produce a number of different products. Primarily the focus of many of our products is on clinical effectiveness as well as cost effectiveness. We do a number of different things, but for today, I will be focusing on both clinical effectiveness and cost effectiveness. So in terms of clinical evidence, and I'm sure this has been a topic that's been discussed with respect to the meeting topics that you've had, this tends to be quite a focus for us. So whether or not technologies work, how they work, and it's a critical input for the economic component when we're actually doing economic evaluations and economic models. Our focus tends to be on RCTs as they represent the highest quality evidence. They tend to be a gold standard. Their experimental design means they have strong scientific rigor. They have a strong experimental design so that we could actually test certain hypotheses in terms of whether an intervention works compared to a comparator for a specific outcome. And as a result, there's strong internal validity in that we can actually prove that there's some causality associated with one intervention over another for a very specific outcome. But when we're thinking of using information at a broader scale, from RCTs, sometimes there may be some limitations. So they often tend not to be comparative. In our case, we do have a drug review program, and when we're looking at drugs coming to us, it's typically very shortly after it goes through regulatory approval. They tend to be placebo-controlled, so not necessarily looking at treatments that we would be most interested in considering. And often they're only single, they're versus one comparator, so we're not necessarily understanding what the drug looks like compared to all comparators of interest. In addition, there's interest to get drugs to market relatively quickly, so they tend to be short in duration. It's not uncommon for us to review drugs with trials of 8 to 12 weeks, so it's hard to know what happens after to patients. So we're not necessarily getting the outcomes that we're most interested in terms of final outcomes, final endpoints, patient relevant outcomes, things that are clinically meaningful, but instead we tend to look at surrogate outcomes, intermediate outcomes, as well as potentially biomarkers. And the RCTs are typically conducted in very specific patient populations. And so if we look at the clinical trial protocols, there's a long list of exclusion criteria. The trial designs themselves, sometimes they're looking at enriched populations or specifically have long run-in periods in order to find responders, but these aren't necessarily the patients that we would actually like to use excuse me, the treatment in. And so when we're thinking about making decisions at a broader scale, the results of clinical trials aren't necessarily very informative in terms of how the drug will work in actual practice, largely because they're designed to answer a very specific question. And so when we're trying to make decisions, RCTs alone may not be entirely informative. So we still might have questions despite the RCT or systematic review of RCTs regarding whether or not a new technology would be better than what we're currently doing. We might also want to know how we should use the technology to maximize benefits of patients or as well minimize harms. And we might want to have a sense of what the value this technology brings to the healthcare setting or system. And so that's where decision analysis comes into play. And so decision analytic models is a framework whereby we would bring together different pieces of information, such as information from RCTs or systematic reviews on the treatment effect, information on the natural history of the condition, resource use associated with treating the condition as well as administration of the treatment. And then it would allow us to really bring all these pieces together and predict what the implications could be when tying all of the information together. And this tends to be a very powerful tool in that it allows us to do experimentation that may not be possible in the real world. So often when we're running our CTs, if we want to make changes or look at different patient populations, it requires amendment to protocols, additional recruitment of patients, so it takes longer. These aren't always possible, whereas within a decision analytic framework, we're able to conduct a lot of what-if analyses, pulling together information from various sources. It also allows us to extrapolate beyond the time horizon for which we have data. So often when we're looking at very short-term clinical trials, we can consider what potentially could happen with respect to treatment effect over time. So exploring whether or not there's any changes in durability of effect, what that means for the value of the therapeutic that we're looking at, or maybe if dose escalation is required over time, again, what does that necessarily mean when we're thinking about making decisions? And it allows for this assessment of value for money. So what are we getting in terms of population health when we actually spend and consider using these treatments within our budget? These models also inform us in terms of what really drives results and what areas for which we look at, do we have high uncertainty associated with some of these parameters in the model? And this is helpful because it allows us to understand where potentially future research could be very informative. So we might be making decisions, there might be uncertainty associated with these decisions, and there may be areas in which additional research would actually help us to understand which patients should we be using treatments in, or whether or not we should be using it, or should we be using it more. So health economics uses decision analytic methods. So they tend to be based on decision analytic models. And so by doing this, we can compare a technology or a drug with relevant comparators, so all relevant comparators, so we could get a bigger sense of how does this look like compared to current management. And by doing so, we can understand the opportunity costs with adopting new technologies compared to what we're currently doing. And what we do within these models is we consider the total cost associated with the new technology. So that would be the treatment cost as well as administration costs if there are any, as well as management of the condition. And we would compare that with all relevant comparators that comprise current management. Similarly, on the effect side, we'd be looking at the effects of the treatment, what it does in terms of management of the condition, whether or not it delays progression or it results in symptomatic relief, as well as any issues with respect to adverse events. And again, we would compare those effects with comparators. And this is looked at in terms of a ratio. So we would compare the incremental total cost with the incremental effects. And this is what we would call an incremental cost effectiveness ratio that you might see in publications. The effects can be reported in terms of clinically meaningful outcomes, and typically we convert them into a common metric, what we call a quality adjusted life year. And this allows us to compare different technologies within a condition or different technologies across different conditions. So why consider health economics? And it's really the issue of constrained budgets and having to make choices. And so there's some issues with respect to what should we prioritize when we have to make choices, and what information do we bring to the table? How do we prioritize? What are we getting? What are we foregoing? So this makes it a very useful tool for decision making to understand what the potential implications are for making really tough choices. So what happens in rare conditions? So often there's the question being posed, should we consider health economics? Does it really matter, given the fact that we know that these are very expensive technologies? Maybe the question really is, does rarity or need preclude the need for the assessment for value for money? And maybe do these treatments warrant a premium over other treatments? So these are all really good questions, but I think they're still answered by using this common framework. So what are some of the issues when we get into rare diseases? So typically, there are very few patients with the condition. So sometimes there's the question of there's only a small number, we could probably afford it, so maybe it doesn't really matter if we look at the economics. Similarly, there may be very few therapeutic options available to patients. And so in the absence of having a new drug technology, we might be left with medical management only. There's also the issue of the identifiable patient, and often because these conditions are rare, we do hear about the patients that are affected, and we know who would be affected given the decisions that we have to make, and that makes it challenging and quite political as well. But the challenge here as well is that there's greater uncertainty. In some cases, just given the rarity of the condition, there's less known about the clinical pathway for patients. There's also potentially less known about the treatment itself because the trials tend to be small. They might not be, again, measuring outcomes of interest. They might not be conducted over a long enough period. So there's uncertainty associated maybe with the condition, but also as well regarding the treatment effect. And because we're dealing with a limited budget or a fixed budget, there's going to be winners and losers associated with any decision we make. And we won't know when we choose to fund one drug over another, who might be losing out. So it's easy to know maybe in cases of rare conditions who might be gaining. But again, when we make these decisions in the absence of a more structured framework for considering how we make decisions, we may not really know who that unidentifiable patient is. And probably the biggest issue in this space is the high cost of treatments. And if these treatments weren't as expensive, we probably wouldn't have the same level of discussion around them, but they are very high cost. So economics does provide us with a consistent framework. It allows us to understand how we go about making decisions. And so we still have to make these tough decisions, but at least we're making them on a consistent basis. We know what we're giving up and what we're gaining across different condition areas. We can also understand the impact of uncertainty. And so often there's some uncertainty with respect to which patients these treatments may work better in. There might be some subgroup analyses, but typically they're very small as it is. So we can't really do a lot of stratified analyses, but they allow us to explore and conduct what-if analyses. And so we might have a better sense of where we might want to do further studies in terms of different patient populations. And again, as mentioned, it does represent a tool whereby we can identify these evidence gaps and we can understand where future research would be most helpful. So I'll just provide a very high level example just to illustrate the point of where this might be helpful and why it might be helpful. So we can consider, excuse me, a situation where we have two new drugs, one drug for a common condition, as well as one drug for a rare condition. For our setting, the common condition affects about 100,000 patients, whereas the rare condition affects 10. The cost of treatment per patient per year is $100 for the common condition and $1 million for the rare condition, so not very uncommon from what we might see. So both result in the same annual budget, $10 million. And suppose someone were to provide you $10 million of additional budget annually, really you could afford either. And how would we make this choice? And there'll probably be a lot of discussion around that. You know, the 10 patients may have very few therapeutic options. The condition could be quite severe. We might feel that that might be worthwhile. Typically, when we talk about common conditions, patients do have other therapeutic options available to them, so maybe there's less of a need. So it's hard to make decisions when we just look at maybe the clinical information as well as just budget. So to that point, if we consider both of these treatments using decision analytic methods and looking at the value that each bring to the healthcare system, what we can do is we can produce what we call this incremental cost effectiveness ratio, looking at the common condition or the drug for the common condition versus relevant comparators. And we would do our modeling exercise and we might come up with an incremental cost effectiveness ratio of $20,000 per QALY. So that means we would have to spend $25,000 in order to gain 1 year of perfect life. For the rare condition, we would do the same exercise, and we would find that the drug for the rare condition versus, say, medical management would result in an incremental cost-effectiveness ratio of $2 million for QALY, which means that we would have to spend $2 million to gain one additional year of perfect life. And our assumption for health economics is that it doesn't matter who obtains the QALY, the quality adjusted life year, it's the same regardless of which patient receives it, who receives it, or which condition it's received in. A QALY is a QALY. So again, if we had the $10 million to spend, what would we do? We know that it takes, or it costs less for us to obtain one QALY for the common condition, and then $2 million for the rare condition. So we know that it's better value, but there still might be reasons why we would choose to fund the drug for the rare condition. But what we do know when we think about making decisions is that we would gain more quality adjusted life years with the common condition than we would with the rare condition. That doesn't mean that it's a slam dunk. That just means that the decision maker knows what they're giving up if they're to fund the rare condition. So it's informative in knowing how we make decisions, as well as what the opportunity cost is associated with making one decision over another. And so what health economics does is it provides us this idea or this estimate of what the value is of different interventions. And it allows us to assess opportunity costs. And that's not to say we know where decision makers will go. There's a lot of components that go into decision making, but it allows them a common framework to just understand what's gained and what's lost, and potentially who may gain and who may lose. So hopefully from this very short presentation, you've gained an understanding of why economics is important, why economics allows us to go beyond clinical studies where maybe we need a bit more information to make decisions. It does provide an assessment of value to the healthcare system. It does provide a platform, and we didn't get into this a lot, but to assess the impact of uncertainty. So typically we don't only have a single value, but we have different statistics regarding how likely the value or the estimate is to occur, and under what scenarios would we see this number changing. It also allows us a framework to make consistent decisions, or at least understand what we're giving up and what we're gaining. And it can provide us areas in which additional research could be informative for future decision making. And so with that, just given the level of uncertainty that we often consider, or we're often faced with when we're dealing with conditions for rare diseases, it's a helpful tool just to understand where these evidence gaps are, as well as potentially what's being gained as well as what's being lost.
Great, thank you so much for that wonderful presentation. This is now open. We have time for a few questions. Dr. Griggs.
A wonderful talk. Thank you so much. It was really good. I spent enough time in England that I've been following their decision making, probably more than Canada. One of the things that sort of startled me was when they decided that the quality adjusted life year cost, if you had cancer, was 2 to 3 times, you could pay for something 2 to 3 times higher because you had cancer. And that seemed sort of counterintuitive to me, possibly because I don't have cancer and I don't care of cancer patients. So that's one of the, and this is, it seemed to me more of a political issue, and you would like to keep people like you in control as opposed to, particularly in this country, politicians.
I think that's an excellent point, and I think they've got into a bit of trouble with the Cancer Fund as well. But that's the challenge. It's hard to make decisions. It's hard to make decisions between cancer patients and between patients with rare conditions. And so by having that very standardized framework, at least we can try to make those hard decisions. It's always going to be hard to say no for some of these new therapeutics, especially where we know there's a therapeutic need. But hopefully by doing these exercises, at least it's helpful to actually have a common framework by which we can defend decisions.
There's one question in the chat, Lindsay.
Yes, it says, Karen, could you comment on using general population-derived utilities versus preferences derived from specific populations, for example, rare disease patients?
So that's a good question, and it tends to be a bit complex. So what we recommend at CADIV is that we recommend that the preference is obtained from a general population. And so these are complicated questionnaires, and they tend to be filled out by the general population. But when we're actually looking at them being completed, so for example, some of the questionnaires such as the EQ5D or the HUI, these are population-based preferences, but the questionnaire itself is filled out by patients, right? So when we're actually doing these exercises, they're filling them out, and that represents different health states or different different states that they would experience along their clinical pathway, and that's what we're trying to capture. So while the set itself is based on general population values, what we're trying to get is a sense from them what these specific scenarios look like to them. So agreed, it is a general population-based values preference set, but we're still getting input from patients with respect to what these different scenarios look like from their perspective.
Great. I think we have one more question. Could you talk about how to value a definitive therapy for a fatal disorder in the first year of life that's given in the first weeks of life and results in a normal outcome? Does the modeling break down with this sort of therapy, thinking of the SMA example?
Find the question.
If Russell Russell wants to unmute and answer in person, feel free.
So the idea with these decision analytic models is that it would capture the progression or the natural history of the condition. Sorry, I'm just reading as I'm trying to respond. So in this case, where a patient would progress to a normal outcome, this would be captured in that we would continue to capture the patient as they progress through the clinical pathway. Similarly, in the case where they don't receive the therapeutic and they die within the first few years of life, they wouldn't accrue any further benefit within this modeling exercise, and they wouldn't have those clinical benefits or the associated costs. And so we would still be capturing the clinical the downstream clinical benefits as well as costs that would accrue because they don't die within the first few weeks or months of life.
Great. Thank you very much. I think that's all of the questions we have. So again, thank you very much for a great presentation. So with that, we'll shift to our next presenter. Megan Imerino is from Nationwide Children's Hospital, and she's going to be presenting her project on a novel approach to optimizing movement in treated children with spinal muscular atrophy.
All right, let me share my screen. Perfect. All right, so thank you, Lindsay, for the introduction. And also, I want to thank Muscle Study Group for inviting me to talk about this exciting project. So as you are all aware, with the increasing availability of treatment options for patients with SMA, clinicians are now being presented with new and exciting challenges to our patients' care. These young kids that we're seeing who have been treated are achieving motor milestones and showing impressive improvements in motor skills. However, because of early motor unit loss or the age at which they were being treated, many of them still present with a motor delay. This motor delay not only impedes their ability to achieve new motor skills, but also has an impact on their global development. In general, it affects their ability to participate and function in their current environment, and it impacts their ability to advance future cognitive, language, and perceptual development. As pediatric therapists, this is really why we push for early targeted interventions for our patients, and this is most certainly the case for our patients with SMA. However, our current model of outpatient therapy does have its limitations. Insurance isn't real quick to cover year-round therapies and typically has visit limits. And then depending on where the family lives, patients may not have access to pediatric physical therapy services. They may not have access to therapists who are familiar or have worked with children with neuromuscular disease. And also it may just be too much of a burden to travel for families many times a week or even once a week to an outpatient therapy center. If we want our kids to develop new and challenging skills, they do need to practice. One modality that is commonly used in the neuromuscular rehabilitation, or I guess pediatrics habilitation, is a body weight support harness system. It alleviates some of the burden of their body weight, allowing them to practice kind of new skills safely and within their current level of function. Unfortunately, these are typically heavy, they're very expensive, and they are typically limited to an inpatient or a hospital setting. They only generally allow one plane of movement, so walking forwards or walking backwards, which makes it really difficult for real-world translation of skills. There is an in-home option that is portable, it's collapsible, and it's commercially available. It's an adaptation of a 10 by 10 kind of camping frame that was outfitted with free moving perpendicular crossbars, which allows 360 degrees of near frictionless movement in any direction within the frame. Because of the friction limiting design, even our weakest kids are able to propel themselves forwards, backwards. They can do little spin moves, they can move diagonally, and they can even transfer or transition through position. So they can go from lying on the floor to a kneeling position, they can go from kneeling to standing, and they can really explore on furniture, around furniture, and anything that's within that 10 by 10 frame. And the cool part is that it's very adaptable. You can change the amount of support and the type of support that you provide that consistently challenges the child at the current level of function that they are. So our team thought that these would be great to use with our kids who have been treated for SMA. So we designed a study to explore the utility and the feasibility of using these systems in family homes. We included subjects with 2 or 3 copies of SMN2. They obviously had been treated, received some sort of treatment for SMA. They had to have upright head control, and that's merely like a safety concern. And then they also had to be under 60 pounds. So that's the weight limit of the system. We used the neuromuscular grow, the Hammersmith expanded, and WHO motor milestones as outcomes and assessed them at baseline, 3 months, and at 6 months. And then after their final study visit, we provided families with a survey to report on their overall experience. It had questions about ease of use and perceived benefit of use, including motor skills and non-motor skills such as social interaction, like playing with friends, playing with siblings, or interacting with family members. We originally enrolled 33 subjects. 1 subject did drop out within 1 week of enrollment, and that was due to family issues that were not harness related. And then we are still waiting on the final usage reports from 2 of our families, which was, as you'll see later, it was used for grouping analysis later on, and I'll talk about that soon. So we're leaving them out at this time, and I'm reporting on 30 subjects. This table here shows the characteristics of the subjects who were enrolled. This was a sample of convenience, so there was a range for enrollment age and age of treatment initiation, but the central tendency for age of enrollment was around 2.5 to 3 years. Most of our subjects were under 4 years of age, but we did have a couple who were 6 years and then 1 child who was 9 years of age, but still met the enrollment criteria. Again, a majority of the subjects had been treated at or before the age of 2 years old, and the average time of being on treatment for the whole cohort was at baseline for this study was 1 year. All subjects reported at least 1 hour of exercise-based therapy a week, and even with that, all 30 subjects had a gross motor delay at baseline. So first, looking at those motor milestones, all subjects were able to sit independently at baseline. I think that kind of goes with upright head control, but no one of our subjects was walking independently. At the final visit, we had 19 new achieved motor milestones across the entire cohort. As you can see, crawling, standing with assistance, and walking with assistance were the most commonly gained skills. As for performance on functional measures between baseline and 6 months, overall, all of our subjects either improved or had stability in their Hammersmith and GROW scores across visits. As you can see in the graphs, we also found a significant correlation between their reported frequency of use and improvement in testing scores. Based on the PT literature that's out there and in pediatrics, an average of 4+ hours a week is considered kind of an intensive therapy dosage. So considering this correlation, we then divided the cohort based on frequency of use, with those reporting 4 hours or more of use a week being binned into like the frequent group, and those reporting less than 4 hours a week in the infrequent use group. And then you can see in the table comparing group change scores from baseline to 6 months, we found a significant difference in Hammersmith and GROW change between the frequent and infrequent user groups. But we really wanted to explore what other variables impacted the improvement in functional testing scores to see if there was a more influential factor that we were not accounting for. We reused linear regression modeling to test the significance of possible confounding variables. It's expected from the literature that what we know that the earlier age at treatment initiation results in better outcomes with this group. And so we looked at frequency of exercise, time since treatment initiation, and also age at enrollment. And as you can see, the results show that frequency is the only significant predictor of Hammersmith and GROW change after accounting for other predictors and on its own. In our data set, neither time since treatment nor age at baseline significantly correlated with a change in either outcome score. And overall, families reported positive experiences using the harness system. So as I said, we provided families with a final use or a final end of study survey. And because of COVID, we had to have some mailed out and then mailed back to us. And we've only received 26 of the 30 post-enrollment surveys. So this slide is based on those 26. As you can see, many of the families reported that they believe the harness system improved their child's motor skills and non-motor skills. No families reported difficulty using the system, and a majority of them found the experience beneficial. Families also noted that the harness system increased their child's independence, their confidence, and allowed them more dynamic interactions with their brothers and sisters. There are also many other benefits to using the harness system in any kid with a motor delay and in our kids who have been treated for SMA. It provides an opportunity for dynamic weight bearing compared to the standard issue static weight bearing like standing devices that are typically prescribed to kids. Muscle tension strains from repetitive mechanical loading are so important for bone health and for bone development. Also, kids learn through exploration. This system encourages self-initiated movement, allowing children to explore, to perceive, and to learn from their environments. This interaction with their environment is key to cognitive development, especially in young neuroplastic kids. There's also digestive benefits to being upright. And then also, this is exercise. There's no current consensus recommendations on exercise dosage in SMA. But we do know that the more we exercise, the more we move, the more we use our muscles, the better we feel, and the more we protect against disuse atrophy. There are obviously limitations to our study. We relied on parent-reported frequency of use, so families were given activity logs to fill out. I think future studies could use accelerometers or activity trackers to kind of have a more quantifiable and use of the harness system. And then the design of the harness is kind of a U-shaped onesie that, although it is designed to pull some of the central forces outwards, there were, in our older males, there were some reports of discomfort using this type of harness, or this type of harness, denim harness. And then we also did not control for other interventions. But in conclusion, the body weight support harness system is a safe, it's feasible, it's enjoyable, a family-driven exercise modality that can really optimize function in children treated for SMA. Our data suggests that frequency of exercise optimizes motor outcomes, and since we can't expect parents to be their child's in-home therapist 7 days a week, this harness system is a great facilitation tool for getting the children the exercise that they need. And yeah, I want to thank my team and Dr. Linda Lowe's, who's our PI for our lab, our biostats group, and then Novartis Avexis at that time for their grant support. Obviously, all of the families who agreed to participate in the study. You can, if you have any other questions afterwards, my email is megan.imarino@nationwidechildrens.org, or you could email nmdtrialinfo@nationwidechildrens.org, because that is easier to spell than my last name. Thank you.
Thank you very much, Megan. That was a great talk. We do have one question at the moment from Igita. It's where were there differences in the frequency of use? Did you explore reasons for lower use in that group? Great study, by the way.
Well, thank you. I think that is something that we're going to kind of look into, and thank you. That was that's a good point. That's something that we're going to look into in future analyses, and as we kind of look and get everything back and move forward.
As a reminder, you guys can enter questions in the Q&A section or raise your hand to ask a question in person.
Could I ask a question, Lindsay?
Yes, you may.
I was just thinking, so my understanding is the exercises so far are purely directed at children. I was just thinking, as these grow older and they're transitioning to adult physicians, are some of these things transferable into adulthood as well?
So specifically with this harness system, there is a 60-pound limit. They do have a less portable version that is available for the home, and I know the company that actually makes these does in-home track systems for families as well. So those would be options for our older adults. But specifically for this group, it was there is a 60-pound weight limit. So
thank you.
Thank you.
Any other questions? Okay, great. Thank you, Megan. That was, again, thank you for that wonderful talk.
Thank you.
So now we are scheduled to have a break. We are running a little early, so I'm going to ask our organizing team, would we like to stick with our 15-minute break, or should we plan to come back as planned at 10 past?
I would suggest coming back at 10 past, just give people a little bit longer, just for those that may be tuning in for a certain lecture, don't you think?
Yeah, I think there is a tendency for people to dip in and out a bit more than they would in a real life conference. So yeah, I'd agree with that.
Yeah, let's come back at 10:10.
Perfect. Then we'll take that break and we'll plan to start promptly at 10 after the hour.
All right, great.
Thanks, Lindsay.
Thanks, Lindsay. Yeah. And then James, you're leading the next discussion, right?
No.
No?
I'm from the top of the second column.
Oh, I'm looking at the wrong. Okay. Oh, so Lindsay keeps going. I
Yep.
got
Lindsay
it.
keeps going, and
I got it. Yeah,
it kind
yes. Now I see.
yeah.
Yeah, you're purple, she's green. I got it. Thanks.
Eleonora wanted to test her microphone. So if you're there, we can do that now.
Nice music, Gil.
Thank you.
Very nice.
Eleonora, are you there? She may be away. All right. Back to the music then.
No.
Okay, we're testing Eleanor's microphone first. Your mic is on. You want to give me a sound test?
Hello, can you hear me?
Yes, I can.
Okay, perfect. Thank you.
Okay, great. Talk to you soon.
Mhm.
And then we're going to test Paloma. Ask her to unmute.
Hello, can you hear me?
Yes, I can.
Okay. And you want me to prepare the slides now or?
We're showing a sponsor slide for this during this break, but before, right, a little bit before your talk, we can pull yours up.
Okay, perfect.
Unless you want to test it now, we could do that quickly.
Yeah,
but then
we can do it. Yeah, I'm going to
Okay.
share. Is that okay?
Yeah, that's fine.
Okay, wonderful. Continue. And this one. Do you see it?
Yes, I do. Nice and full screen.
Okay, wonderful.
All right.
Well, thank you so much.
I'm gonna take it down for now, but
Of course.
okay.
What's that?
We're just testing some stuff for our speakers. Okay.
Hello, everyone, and welcome back to the next section of our morning's presentations. So our next presenter is Paloma Gonzalez Perez, who is an MSG Fellow at Massachusetts General Hospital, and she's going to be talking to us about molecular biomarkers in myotonic dystrophy type 2.
Okay. Hello, can I share my screen?
Yes, you can.
Yes, you can.
Perfect, thank you. Okay. Well, hello, good morning. I'm going to present the work that I will be doing for the next 2 years as a muscle study group fellow. And I'm investigating the molecular biomarkers of myotonic dystrophy type 2. And okay, I'm going to start reviewing the common pathogenic mechanism in myotonic dystrophy type 1 and type 2. So as you know, myotonic dystrophy type 1 is caused by a trinucleotide expansion within the 3' UTR of the DMPK gene, and myotonic dystrophy type 2 is caused by a tetranucleotide expansion within intron 1 of the CMBP gene. These DNA molecules transcribe into RNAs, and these expanded RNAs accumulate in the nuclei of the cells, forming what we call the RNA foci. These RNA foci interfere with very important proteins. MBNL is the best studied one in myotonic dystrophy. And this protein is a splicing regulator. So when this protein is sequestered in this RNA foci, it loses its function. And as a result, we have a subset of other mRNAs transcribed from other genes that are not normally spliced. And then the products of these mRNAs are abnormal proteins. So we know that in myotonic dystrophy, we have somatic instability at the level of the DNA. And we also know that these diseases not only affect the skeletal muscle, but other organs, such as the brain and the heart. So the best explanation that we have so far for this multi-organ involvement is actually this subset of not well-spliced mRNAs. So in this sense, we know, for example, that missplicing of the chloride channel is associated with myotonia in these patients, and this is the best well-known association, but there are others in the literature, such as, for example, missplicing of the insulin receptor associated with insulin resistance, or calcium channel associated with muscle weakness, or sodium channel associated with arrhythmias. And this slide is just to show you that in myotonic dystrophy, we may have RNA transcripts that are not well spliced, and we call it missplicing. Basically, something that is there shouldn't be there, or something that is not there should be there. But also, it could be that the isoforms are normal from a qualitative standpoint, but the quantity of specific isoforms is what is abnormal, and this is also pathogenic, and as we will see later. So why we need biomarkers in myotonic dystrophy? So obviously it would be good to have a good biomarker that help us to monitor the response to treatment in future clinical trials. To my knowledge, we don't have clinical trials right now. And also to know more about the disease and also the prognosis of our patients, especially in this multi-organ in this multi-organ disease. It would be good maybe to know which patients are going to be with higher risk, for example, for heart problems. And which type of biomarkers? Well, we favor investigating molecular biomarkers because we know the genetic basis of the disease, and also the pathogenic mechanism is RNA mediated. So, and between the DNA and the RNA, we favor RNA. And I think you all know this. We know, for example, at the level of the DNA in myotonic dystrophy type 1, we know that the length of the expansion correlates well with the age of onset and also disease severity. But we also know, for example, if we focus in the classic form of myotonic dystrophy type 1, which is the most common form we see in the adult neuromuscular clinic, we know that patients with the same number of repeats still may have very different phenotypes. So here in this scenario, the length of the expansion doesn't help to predict prognosis or severity. And in myotonic dystrophy type 2, things are more complicated. So we know that more than 75 repeats is diagnostic for the disease, but we also know that these patients may have very high number of repeats, up to 11,000. And this is very difficult to determine sometimes how many repeats has a specific patient. So for those who have a specific number of repeats, we know that it does not appear to be a correlation between the number of repeats and the age of onset or disease severity, unlike in the M1. So then we go with the RNA. And in this slide, I have tried to summarize the most important data that guide us to continue to investigate RNA as a potential biomarker in myotonic dystrophy. So here in this first table, I have tried to summarize the most important data from these 3 papers and 3 groups. So these 3 groups investigated the transcriptomic changes in muscle tissue from patients with myotonic dystrophy type 1 and myotonic dystrophy type 2. And they came up with a panel of transcriptomic changes in the muscle. So for example, the insulin receptor transcript with the inclusion of exon 11, and this is an isoform, is significantly reduced in myotonic dystrophy patients when comparing with controls. And here controls is not only healthy subjects, but also other patients with other type of muscular dystrophies. And very nicely, Nakamori and colleagues show how these transcriptomic changes in muscle in myotonic dystrophy type 2 patients were more pronounced in vastus lateralis than in tibialis anterior, and more pronounced in tibialis anterior than vastus lateralis in myotonic dystrophy type 1 patients, which very nicely align with the distribution of weakness that these patients have. And then at the level of animal models, the transgenic mice, we know Wheeler and colleagues show very nicely how when they treat with antisense oligonucleotide, targeting the expansion at the level of the RNA, these transcriptomic changes correct. So in animals, the correction of these transcriptomic changes is the best indicator that the drug is working. So hopefully we will have something like that in clinical trials in the future. So that's why we want to continue to investigate the role of RNA as a potential biomarker in myotonic dystrophy. We would like to avoid muscle biopsies, and we would also like to have a biomarker that not only correlates with the muscle weakness, but also with other clinical manifestations of the disease. So that's why we are investigating the extracellular vesicles. So this is just to review what are the extracellular vesicles. I have represented them here in this slide in yellow. It is a mode of communication between cells. They receive different names depending on the size. And these extracellular vesicles have been identified in different human biofluids, in urine, serum, saliva, CSF, breast milk. And the content is a variable. Could be proteins, DNA, lipids, and different types of RNA. Specifically, the RNA has been used in, has clinical applications and that have been already used, for example, as a diagnostic and prognostic biomarkers, especially in cancer, and also as a therapy, because these extracellular vesicles can deliver a treatment without activating the immune system of the patient. So they are very attractive from that standpoint. So what I'm interested to do is investigating this mRNA in the urine and serum of patients with myotonic dystrophy type 2 to see if they are good biomarkers of disease severity. And I'm very lucky because this has been done in myotonic dystrophy type 1 by my mentor and his lab. In 2018, they published this paper in Nature Communication. They analyzed the same transcripts that I showed you before that were seen in muscle tissue. They investigated that in the EVs, extracellular vesicles, in the urine of 40 patients with myotonic dystrophy type 1. And you can see that the changes are consistent in the same direction in the transcriptomic change as it was previously reported in muscle tissue. So then my specific aims will be basically the same approach, but in patients with myotonic dystrophy type 2. I will investigate the transcriptomic changes specifically for these 10 transcripts. I will also obviously look at the CMVP because it's where the expansion is located. And by doing RNA-seq, we may find other transcripts that we are not aware of at this time. And of course, we have to correlate, we have to see if there is any correlation between the transcriptomic changes and clinical parameters. And we are planning to collect these clinical parameters. And we are already using some questionnaires in the clinic, such as the active and some scales to screen for fatigue, daytime sleepiness, and cognitive impairment. So from a practical standpoint, what I do when I have the patient in clinic, of course, we have to obtain the consent. We collect the urine and blood, ideally both. And then we perform first centrifugation, low speed centrifugation. We get a pellet. This first pellet is going to contain the cells. We are not interested in the cells right now. So we continue with the supernatant. And then the supernatant undergoes another centrifugation, this time ultracentrifugation. And we get this pellet. This is where our EVs are going to be located. So then we follow a standard protocol for RNA extraction using trisol, and then we have our RNA. So this sample goes to RNA-seq to identify new transcripts that we may not be aware of, or we continue looking for a specific transcript, such as CMVP, by converting the RNA into cDNA and doing PCR, semi-quantitative PCR first, and then more quantitative PCR as a droplet digital PCR. So, and then here is some preliminary data that I can show you. So obviously we have more patients with DM1 than with DM2. Here you can see 4 patients with DM2. We have urine for these 4 patients. And we have the primers for these 3 transcripts, as you can see here. And then we look for a specific, I mean, these 3 transcripts are the ones that I showed you before in the table, and they were also shown to have differences in muscle tissue. So basically we designed the primers to detect that, and you can see that the group of myotonic dystrophy type 2 is in an intermediate zone between the unaffected group and the DM1. Obviously, you know, low number, we cannot say anything. We need to continue recruiting patients. This is in urine, and then I can show you more or less the same in serum. For serum, we only have 3 samples from 3 patients, and you can see maybe a little bit of difference there, but we cannot make any conclusions right now. And this is for these 2 transcripts that also were in the table that I showed you before. So now, okay, now coming back to the table. So I can show you, I'm just adding what I just showed you, and you can see the direction of the transcriptomic change is consistent with what it was seen in muscle and also in the urine of DM1 patients. Of course, you see many blanks here that I hope I can fill in the next 2 years. So, and then I think this experiment, next experiment is very interesting. So we look also CMBP. We know from a prior publication, Snyder in 2018, show that intracellularly and in muscle, in other tissues too, but in muscle, intracellularly, they saw that this isoform containing intron 1 and expansion is increased in patients with myotonic dystrophy type 2. So then we say, okay, how it's gonna look in the EVs? And then we wanted to detect the normal splice form of CMBP. So basically we design a primer here that spans across the splice site between exon 1 and exon 2 and the reverse primer. And obviously this PCR is gonna detect the normal form of CMBP in the EVs. And we use our urine samples. We have 4, and we still, it's a low number of patients we know, but I think the difference is quite promising. So the normally spliced CMBP is significantly lower in DM1, in DM2 patients when we compare with controls. And this is what we have so far. So I hope I can show you more in the next meeting. And I just want to say thank you to the patients and families, and everybody in Wheeler's lab, and especially to Thurman Wheeler also for all his guidance and mentorship with this project. Everybody also in the clinic, because obviously we have patients who are followed by different providers and they are contributing very actively. And also Sudeshna Dash, she's helping with the bioinformatics. And our collaborators, we are very fortunate to have them on board, Dr. Amato, Dr. Pubanat. We are right now establishing, in the process of having IRB approval to collaborate with Dr. Pubanat and also Dr. Darvas. And also thank you to the Myotonic Dystrophy Foundation, because they gave me the opportunity to explain this research to patients with myotonic dystrophy type 2. And of course, the funders, Muscle Study Group, AAN and ABF for supporting me and the project. Thank you.
Thank you very much, Paloma. That was great. And just as a reminder to participants, you can raise your hand to ask a question in person or enter any questions in the Q&A or chat sections. And it looks like Zhao Shenlu, you have your hand raised.
That might be a mistake. He had his hand raised yesterday and didn't have a question.
Okay.
Oh, let's go. There we go.
Do we have any questions for Paloma?
So this is Rick. Maybe I missed it. How many subjects do you need for this study this year? How many samples?
We are planning right now in the clinic. We have in our institution, we have 15 patients that we are planning to offer. I mean, 4 of them are the ones that I show you. We have 3 serum samples and 4 urines. Sometimes it's difficult to get the serum because we don't have the nurse with us. But we plan at least the 15 that we have in our institution, but we hope more. I didn't do any power.
Yeah, that's, I guess, the question. How many do you think you need? Ideally.
Ideally 40 as in the M1, as the study that they did, I think.
Yeah, I mean, if that's really the issue, you can get that with help from the MSG. Just we'll volunteer to help you. I mean, I guess the idea is if you need samples, all you really need to do is reach out and we can get them for you.
Okay, thank you. Yeah, I will contact for sure.
Let you know. Yeah.
Any other questions?
That's very exciting. Thanks, Paloma. And are you just starting the fellowship now?
Yes, in July.
Right.
Yes.
So this is the beginning of, and are you going to do it for 1 or 2 years?
I think it's 2, right?
Yeah, no, we funded you for 2, but you said what I'm going to do in this year. So that's what it confused me a little bit. So you have 2, yeah. So, yeah, no, the money's there for 2 years. Don't worry.
Okay.
Okay, great. Thank you very much, Paloma. And with that, we'll shift to our next presenter. Jennifer Weinstein is going to present from the University of Rochester on results from a national cross-sectional study of disease burden in ALS, results from the national cross-sectional study. Hi, everyone. It's a pleasure to be presenting to you today. My name is Jennifer Weinstein, and I'm sharing results from our national cross-sectional study of disease burden in ALS on behalf of Dr. Heatwell's Clinical Outcomes Laboratory. Our national cross-sectional study of ALS patients were members of the CDC's Prevention National ALS Registry. Patients participated in an online survey collecting demographic data and relative importance of 189 potential symptoms.
Before you get into it, I'm sorry to cut you off. Your mic is kind of cutting in and out. May I ask what microphone you're using?
It's just my computer microphone.
Yeah, really, it's like, it's weird. Maybe if you just put your mouth closer to it. I'm not sure.
All right, we will try this.
Yeah, that's better.
I think that's better, yeah.
That's better? Okay, fabulous. All right. So this diagram illustrates the process by which the symptoms of interest in the cross-sectional survey were derived. Beginning on the left, we had 15 in-depth patient interviews that were conducted capturing 762 quotes. These quotes represented 303 symptoms across physical health, social health, and mental health in additional ALS symptom domains. The cross-sectional survey builds upon this prior work to provide the depth and breadth to understanding patient disease burden. So 532 ALS patients logged on to our cross-sectional survey for a total of 497 completed surveys. Our survey participants were predominantly male, aged 32 to 87. The average month since diagnosis was 36 months, with 52 months the average time since onset of weakness, representing a diagnostic delay of 16 months. 20% of patients required the use of a feeding tube, and 8.6% had genetic testing positive for a mutation associated with ALS. The most common mutation that we saw was C9orf with 26%. So examining the distribution of types of ALS from our cross-sectional survey shows that 87.3% had ALS, 5.4% PLS, 3.2% PMA, 2% ALS with frontotemporal dementia, and 0.6% with Kennedy's disease. The breathing status of our participants indicates that 62.8% did not need any assisted ventilation, and only 1.8% used a tracheostomy and ventilator. The remaining 35.4% used BiPAP. Our data on ambulatory status shows that the majority of participants, 38.2%, primarily use a wheelchair or motorized scooters, followed by 31.8% of participants who can walk independently without assistance. This graph shows the juxtaposition of symptom prevalence, which is indicated in light blue, and symptom relative importance, which is in dark blue. The most common symptoms experienced were inability to do activities, fatigue, problems with your hands or fingers, limitations with mobility or walking, and decreased performance in social situations. But this graph importantly reveals the distinction that symptoms of the highest prevalence are not always the symptoms posing the greatest impact to patients' lives. So inability to do activities was shown to have the most importance to patients, followed by mobility. Data on symptom prevalence of ALS themes are used to derive rate relationships across demographic variables. As seen in this table, there are high levels of correlation between the prevalence of our symptomatic theme and our patients' ability to work. The highest correlation between those who work and those who are unable to work is seen with choking or swallowing issues. Our cross-sectional data has allowed us to generate and validate a disease-specific patient-reported outcome measure for ALS that measures overall disease burden and 15 areas of symptomatic burden. Psychometric testing and beta testing phases are now complete, and we're pleased to announce that the ALS High is now available for use in clinical trials. On behalf of Dr. Heatwell's Clinical Outcomes Laboratory, I want to conclude by thanking our co-authors and all of our study sponsors for their commitment to our work. And my team and I are happy to take any questions at this time. Great, thank you. Any questions for Jennifer?
Maybe I could ask one, Lindsay, before this jump in.
Absolutely.
Thanks for the talk, Jennifer. That was great. I just wonder, so what would be the benefits of your tool over the existing tools that are available already? You're able to summarize that?
Yeah, so our PROs are disease specific, so there's a much higher level of sensitivity and specificity as to what we're measuring. And so that is highly preferable over just a general measurement of quality of life or functional status. And we find that in our beta testing, our patients who are taking these surveys find that the level of specificity is, in fact, enables them to really have greater input in, allowing us to understand the status of their emotional and physical well-being.
Great, thank you.
Could you, Jennifer, could you also comment on how you would access this tool or use of this tool in the trial? So that, I guess Dr. Heatwell would have to speak more specifically to that, but it can be licensed for use in clinical trials. It can be used as a health index by clinicians. And for our cross-sectional survey, we implemented that online using REDCap. So hopefully that answers your question. And I see Chad Heatwell has his hand raised.
Good morning, everybody. Thank you, Jennifer, for presenting that data. Just to kind of respond to those last two questions, there's a lot of ALS tools out there. ALS FRS is probably the most commonly used, but this is a pure patient-reported outcome measure. And we designed this from the ground up to really satisfy FDA criteria for use in drug labeling claims. So it's multifactorial. It has capability and sensitivity to measure a lot of different symptomatic domains. So I think it provides a little bit different input, and we design it really to be as responsive and optimal for use in clinical trials. And we want this to be used openly by academic researchers, and just reach out to me. It's distributed through the University of Rochester. So if you contact Jennifer or I, we can put you in the right direction.
So it looks like we have another question. What is the range of scores on the ALS high, and what change in score do you regard as clinically meaningful to patients? Oh, I think Dr. Hewill is going to take that one, if he's still there.
Yes, so to be determined, we've developed about 70 of these instruments, and the MCID for almost every one of these instruments developed through longitudinal studies and anchor methods and statistical methods is about 8 to 9 points. And so I think that we expect that will be similar for this particular instrument, and that data can be used to help design trials as need be. And we literally just finished this instrument within this month. And so some of the analysis, post-production analysis that we're going to do is known groups validity testing to really look at the magnitude of difference between groups with different severity of diseases. And so that will also give us an idea of what's an important difference to measure using this instrument.
Great. Any other questions?
I think one came through on the chat, Lindsay.
I was wondering if you also looked at caregiver burden.
Oh, I can take that. No, we have not yet implemented a caregiver version for ALS. We have done that for several other neurological diseases, but not yet.
Great. Okay, there are no, one more question just came through. So to Dr. Amato's point yesterday, is there weighting or anything to demonstrate that a 1.5 point change type scenario could be proven significant? Go ahead, Chad. I see you.
Yeah, Chad.
Can you hear me?
Yes.
Oh, yeah. So there is a weighting system, a sophisticated weighting system that we include in all of our instruments. And this essentially allows us to prioritize the symptoms and actually the subscales, because the total instrument is derived of many different subscales that individually measure the concepts that are most important, or symptomatic concepts that are most important to patients with ALS. And so we do have a weighting system. You know, a lot of scales use a 1-to-1-to-1 weighting system, which was heavily flawed, because we all know that certain things and certain symptoms really are not equally important to patients. And so we do have this data to back it up from our 500 patient sample about which symptoms are most important and which symptoms really should be prioritized in this population. So we do have a weighting system. And then as far as knowing what's clinically meaningful, we do have mechanisms to really determine that as well. There's a couple different ways to do that. So one way is to longitudinally track patients with this instrument and do anchor methods to determine the MCID, determine the minimally clinically important difference of the instrument in each of the subscales. There's also statistical ways to do this. And the third way that we do this to kind of validate what a meaningful change in the score is we look at different groups of ALS patients in a cross-sectional fashion. So for instance, those who've lost the ability to talk, or those who are on disability, or those with a longer duration And also we look at all other aspects too. We look at age and gender, and we look at genetic manifestations of the disease as well, or occurrence of genetic ALS. And we compare scores across groups that are thought to have more or less disease burden. And so in a cross-sectional fashion, you can determine, and what happens is the ALS high can differentiate between these groups. And we look at the difference in scores in both the subscale scores and the total scores to get an idea of what's the difference in a population of ALS that's disabled versus not. And so it's a complicated question, but there are a couple different ways to really ensure that a certain change in this instrument is really meaningful to patients.
Great, thank you. Okay, and with that, I think we're going to shift to our next flash presenter. Daniel Kalam is going to talk to us from Baylor Medical College about exome sequencing, identifying novel candidate genes, and phenotypic expansion in a neuromuscular cohort.
Great, thank you very much. Let me share my screen real quick. All right. Well, hi everyone. Thank you very much for giving me the opportunity to speak about my research here today. My name is Daniel Callum. I'm a child neurology resident at Baylor College of Medicine. I'm in our research track, and so I'm working this year with Jim Lepsky and Davut Pehlevan, investigating the genetic basis of neuromuscular disease by performing exome sequencing in a large neuromuscular cohort. So I probably needn't tell you all that we've discovered a ton of neuromuscular disease genes, over 500 to date, which affect virtually every aspect of the neuromuscular axis. But despite all these discoveries, somewhere between 40 to 75% of our patients that are felt to have a genetic disorder lack a molecular diagnosis, which clearly suggests that there are many genes and genomic mechanisms of disease that we have yet to discover. So we've been recruiting families with neuromuscular disease for quite some time. We have 64 of them at present. They are all undiagnosed despite prior testing. Patients that are coming from UT Houston, Baylor College of Medicine, Texas Children's Hospital, or international collaborators. And for all these patients, we're trying to apply exome sequencing, ideally TRIA with the parents tested as well if they're available. And then we run this data through our in-house pipeline to analyze and identify candidate variants. This is preliminary analysis of this cohort. I have some exome sequencing data from at least 44 patients. You can see that at present, about half the cohort remains unsolved. 18, or about 41%, have variants in known disease genes that are thought to explain the phenotype. And then there are 4 families for which I've identified novel candidate genes. So today I'm going to talk about 3 of the candidate genes and very briefly about one example of phenotypic expansion that I think is very interesting. The first candidate gene is called collagen 19A1. This was identified in 2 families with arthrogryposis multiplex congenita, as well as proximal weakness, facial weakness, and or diminished or absent reflexes. You can see these 2 patients have rare damaging variants in collagen 19A1. And we think this is a really great candidate gene because in the zebrafish, genetic defects in collagen 19A1 or morpholino-mediated knockdown leads to defective motor axon migration to the neuromuscular junction, which clearly is a mechanism of disease that can cause arthrogryposis. The second candidate gene that I'll talk about is called DHX9. This is a DNA RNA helicase. It's potentially an axonal CMT gene. Initially identified it in a middle-aged woman with axonal CMT. It's a de novo variant in her case. She has no family history of CMT. The variant is quite rare, predicted damaging. And then subsequently, I queried the Baylor Genetics Clinical Exome database and found potentially a second case, again, a gentleman with axonal CMT and a rare heterozygous damaging variant in this gene. Unfortunately, the patient is adopted, so we don't have any family history to work with here. Curiously, both of these variants are found within the same domain of this, the protein product of this gene, very close to one another in an area that's required for the helicase functioning of this, this protein. So we're very intrigued by it. Additionally, DHX9 is very similar in terms of its biological function to a known axonopathy gene, syntaxin. Syntaxin acts to resolve R loops, these DNA RNA hybrids that form during transcription. And any perturbation of this can lead to problems, like genomic instability, problems of RNA splicing and gene regulation. And so we think this is a great candidate. The last novel candidate that I'll talk about is a gene called ENTPD1. This one is not completely novel. It was described previously in two families, well, two families and then one or two since then. But really there hasn't been any functional characterization of these variants. And so the story still is a little, not the firmest association to date. And so I think this family, as well as some of the other ones we've subsequently identified, are going to really help to establish this disease gene association. You can see our family here. It's a consanguineous family with one affected child with complex hereditary spastic paraplegia. They have a very interesting variant in this gene. It's a 2-base deletion insertion that is homozygous in the proband. It leads to a non-conserved amino acid substitution, which is predicted to be damaging. And if you're curious about what this gene does, I've this figure on the right that shows it's a cell surface enzyme. It's fairly ubiquitously expressed amongst tissues, and it acts to break down ATP into ADP and then ultimately AMP, which in turn is broken down into adenosine, a very important signaling molecule in a lot of biological processes. We took this gene to Gene Matcher, and we were able to find quite a few other cases that haven't been reported. A total now of 6 families, I think about 10 affected individuals. You can see the variants they have here. They're all homozygous or compound heterozygotes for ENTPD1. They all have complex HSP. And so we're performing deep phenotyping along with our clinical collaborators, and also working to establish a collaboration to actually test the functioning of these variants in vitro in a lymphoblast assay. So I think this will really help to establish this gene as a cause of complex HSP. And then I don't really have time to go into all the different phenotypic expansions that have fallen out of this work so far. I just wanted to highlight one, the family you see in bold here. This is a patient at Texas Children's Hospital that I've had the pleasure of seeing with a very severe congenital myopathy. And so upon research reanalysis of his exome sequencing data, I noticed that he has a homozygous canonical splice site variant in a gene called TNNT3. TNNT3 encodes troponin T, which is found in fast twitch muscle fibers. It has previously been associated with autosomal dominant distal arthrogryposis. And what I think is happening here is, since you have 2 hits to the gene instead of just 1, and with a loss of function variant, I think that's what's causing the more severe disease. And in line with this, there's been one other case of this reported a couple years back of a patient with Niemelin myopathy and distal arthrogryposis who had a homozygous splice variant in the same gene. And as you can see in the bottom right, they very elegantly rather, demonstrated that there's a loss of this gene's protein product in this patient's smooth muscle, which again fits with the hypothesis that biallelic disease is much more severe than monoallelic disease. So in conclusion, just from our preliminary analysis, we've uncovered a total of 4 novel candidate neuromuscular genes, 3 examples of phenotypic expansion. And I think really in conclusion, I'd say that exome sequencing remains a very powerful tool facilitating the discovery of novel neuromuscular disease genes. And that's it. If you have any similar cases or interested in collaborating with us, do email me at daniel.callum@bcm.edu. That's it.
Great, thank you, Daniel. And it doesn't look like any questions came through right now. And in the interest of time, I think I'm going to move on to our next presenter. So if you have questions that you didn't put into the chat, please feel free to contact Daniel.
Thanks.
So our next presenter is Senda Ajrudris, who's going to talk to us from the Northwestern University Feinberg School of Medicine on a safety study of weekly steroids in muscular dystrophy.
Hello, everybody. Thank you for having me. I'm trying to share my screen. Here you go. Well, again, on behalf of all my co-authors here, I would like to thank the organizer for allowing us to present, trying to push the bar a little bit lower so it doesn't obliterate my slides. Here we go. So I really don't need to emphasize to this audience the importance of corticosteroids in boys with Duchenne muscular dystrophy. However, the benefit of corticosteroid has not been fully investigated in Becker muscular dystrophy or in other LGMD subtype. Would like to present today the preliminary result of our open label safety and efficacy clinical trial of oral weekly corticosteroid in adult patient with Becker or limb-girdle muscular dystrophy subtypes. Really, the idea to use weekly oral steroid in our population came from the preclinical study in the MDX mouse and in limb-girdle muscular dystrophy type 2B.
Sorry, Senda, 1 second. We're only seeing the first slide of your presentation. Maybe you have to, yeah, so maybe stop share screen and try it again.
Okay, sorry about that.
It's okay.
One second.
There we
Do you
go.
see it now?
Yep.
I have to advance the other way, actually. Sorry, it's... there you go. You can see it now?
Yes, thank you.
And is it advancing?
Yes, it is.
Okay, perfect. Sorry about that. So I said really the idea of using weekly steroid came from the preclinical study in the MDX mouse model and in mice model of limb-girdle muscular dystrophy type 2B and 2C, where once weekly steroid was actually superior to daily steroid in improving strength in these mice. And it's interesting that these effects seem to be also circadian, as the mice were dosed in the morning, and that's equivalent to the evening for the adults, for the human. So we actually did right that. We took our data from the bench to the bedside and started our cohort of patients on weekly 1 milligram per kilo of steroid versus, 0.75 to 1 milligram per kilo, depending on their weight. And we asked the patient to take it in the evening after their evening meal once a week. We measured strength assessment, we did muscle MRIs, whole body DEXA scans, and also safety and research lab. We did that at baseline and at 6 months, and we followed them monthly through phone calls. We wanted to recruit more patients, but unfortunately, because of COVID, we recruited 20 patients. Their ages were between 18 and 60. About 70% of our patients were male, and 40% were ambulatory, whereas the rest were transitioning or wheelchair bound. As you can see, the dose of steroid wasn't that high. The maximum was below 90 milligrams per week. We enrolled a very heterogeneous group of patients, and it's unfortunate that we could only enroll one Becker muscular dystrophy patient. Again, I want to remind you that this was mainly a safety study. We were a little bit worried about other previous study that actually showed negative effect of steroid in certain subtype of LGMD. So we were really focusing on the safety, and we can see that the blood pressure, the heart rate, the respiratory parameter, and even the weight were stable. patient lipid profile also remained unchanged over a 6-month period. And although there was trend toward increased hyperglycemia, their hemoglobin A1C remained in the normal range after 6 months. We were, we saw that the prednisone weekly, did significantly decrease their CK level, but we knew not to get very excited about this, as this was shown before without necessarily any kind of functional effect. This change in CK was also reflected in the drop of their ALT and AST. We did DEXA scans to monitor bone density. We did not see any significant change in bone density. However, we were nicely reminded that the use of DEXA could be a good tool to evaluate lean mass versus fat mass in these patients. As you can see it clearly, the red represent muscle, the yellow is the fat mass, and we see a clear difference between the ambulatory patient, the patient who are transitioning to wheelchair, and the wheelchair-bound patient. It's also interesting that actually the weight of these patients did correlate nicely with the weight on DEXA. So for the non-ambulatory patient that are sometimes difficult to weigh, this may be a nice way to determine that. Again, we were nicely reminded that DEXA could be a good tool to evaluate longitudinal change in their muscle mass. This is one of our kind of more spectacular example where we could see the change in the legs and in the arm. The total body mass of this patient decreased because he decided to be in a clinical trial to adopt a healthier lifestyle and start dieting. So this significant change that we saw in the lean mass in the upper extremity did have some functional correlate with some trending towards improvement of their grip strength, and also of their 6-minute, for the lower extremity, of the 6-minute walk and the 10-meter run. However, this should be interpreted with caution as our sample size was very small. We used muscle MRI to determine the fat fraction, and we were thinking that this will give us an idea about the change in fatty remodeling of these muscles. All patients had an increase in T2, and after a 6-month period, we did see the decrease in water T2. This was also shown in other MRI study of Duchenne muscular dystrophy patient as an effect of steroid. I also want to highlight here that it's, since we measured the fat fraction through MR spectroscopy, how difficult it is to do this study in our patient. You can see here that for this patient, for example, the only muscle left is the tibialis posterior, and it's very hard to put the marker there. So our MRI data is still being analyzed, but I want to show you here that, for example, for this patient, this is his baseline scans, and these are the 6-month scans. We did these, every dot on the curve is a slice of MRI, and if we follow these slices throughout the leg of this patient, the thigh and the leg, we do see a decrease in the fat fracture in the thigh muscle. And we can actually see that better here. These are our ambulatory muscle patient, where we looked at every single muscle in every patient being its own control. And the muscle of the thigh seemed to have the most decrease in fat fraction compared to the leg. We don't know if this is just a progression of the disease versus actually a negative effect of steroids. So we are still looking at this data. It is unfortunate that in the non-ambulatory also, it's not as surprising because they don't have a lot of muscle left in their legs, that this data is not as clear. So I hope that I convinced you that using steroid at the dose of 0.75 to 1 gram per kilo per week in a patient with limb-girdle muscular dystrophy is safe and well tolerated. I understand that our study is not powered for efficacy, but it does actually, there is some indication that there must be some increase in lean mass in DEXA and maybe some functional improvement that definitely needs to be looked at in more detail. And we're thinking that a bigger study and also a dose finding study may be warranted in the future. I would like to thank my co-author on this study, and I really would like to highlight one in particular, Aaron Zlokovich, which is a PGY1 neurology resident right now. He did this work, most of it, as a medical student last year. Thank you.
Thank you very much. And it looks like we have time for one quick question talking about the, you included a broad range of LGMD subtypes. Would you have expected different responses based on the type of LGMD?
We were a little bit worried about the LGMD 2Bs because there was a small study in this Ferlin patient that showed a negative impact. As you know, these diseases are very, very rare. So it's very hard for one single center to have a large proportion of each one of the limb-girdle muscular dystrophy. So basically, we just took what we had. But of course, I mean, this created a lot of heterogeneity in our study, especially on the MRI. As you can know, every single limb-girdle muscular dystrophy has a different muscle involvement. And that's the headache that we have to interpret all our MRI data. It's interesting that we found that even within one limb-girdle muscular dystrophy, the 2L, certain patient had complete opposite muscle involvement.
Thank you very much. Okay, so with that, I think I will shift to James to introduce our next presenter.
Thanks, Lindsay. There were a couple of outstanding questions there. Apologies for that. I think in the interest of time, we'll have to move on, but please feel free to contact the speaker in the meantime. So I'm James, a Lillquit neurologist from Manchester in the UK, and it's my pleasure to introduce Eleonora D'Ambrosio first, who's talking about AAV gene therapy for TNNT1 associated Nemelin myopathy. So hopefully, Eleonora, you're there and are able to share your screen.
Yes, can you hear me okay?
Yep, loud and clear.
Can you see the screen now?
Uh-huh.
Yeah? Okay, good. Right. Hello, everyone. Thanks for having me here. My name is Eleonora. I'm one of the neurology residents at the University of Massachusetts. And for the past few months, I've been working in Miguel San Esteves' lab in the gene therapy department on AAV gene therapy for TNNT1 associated Nemelin myopathy. Let's start with some clinical aspect first. Can you see the second slide okay?
Yeah, that's there, yeah.
Perfect. Okay. So Nemelin myopathy is one of the most common forms of non-dystrophic muscle disease with a frequency of 1 out of 50,000 newborn. The term Nemelin actually comes from the Greek word nema, that means thread or rod, because rod-like inclusions are seen within the sarcolemma of muscle of patients affected by Nemelin myopathy. In its most common, which is also the most severe form, patients present with early onset limbs, neck, and truncal muscle weakness, hypotonia, tremors that can be mistaken with seizures, spasticity, skeletal deformity, as we can see here, pectus carinatum deformity in these patients, and inevitably respiratory failure and death. There are many different mutations associated with this disease, mostly are autosomal recessive. But in my lab, we focus on TNNT1, which encodes for the slow skeletal muscle isoform of troponin T. A nonsense mutation in exon 11 of this gene results in selective atrophy of slow twitch fibers, so type 1 myofibers only, and in overexpansion of type 2, so fast twitch myofibers. This type of mutation leads to an early onset fatal form of Nemelin myopathy, which is called Amish Nemelin myopathy. There is no cure for this disease. However, we were thinking that AAV gene therapy would be viable. First of all, because the TNNT1 protein is very small, is 32 kilodalton, and so it is the gene. And then we have now many AAV capsids, AAV8, AAV9, AAV74 that are capable of broad muscle transfer. The problem that we were having was to restrict the expression of TNNT1 to slow twitch type 1 myofibers only. So we were trying to prevent the expression of TNNT1 to fast twitch fibers or myocardiocytes because of the deleterious effect that this would have. So we achieved this with the technique of the microRNA detargeting. I'll explain you a little bit how. These are some preliminary results that my lab collected. So first of all, we decided to test the microRNA detargeting on fast twitch fiber. We decided to take a muscle, the gastrocnemius, which is predominantly a fast twitch fiber muscle in mice. And we generated a few AAV GFP vectors carrying different microRNA cassettes that I named here A, B, C, and D. We injected 6-week-old C57, so wild-type mice, intramuscularly with these vectors carrying different MIR-T cassettes. And then the muscle, the gastrocnemius, was analyzed for GFP expression 4 weeks after the injection. Those are some immunofluorescent imaging data. And so you can see clearly that compared to cassette A and B, where the GFP is fully expressed still, cassette C and D resulted in an overall reduction of GFP expression, meaning that in this muscle, which is again a fast twitch fiber muscle, we have achieved a detargeting of the fast twitch fibers with the vectors carrying GFP. Then, and this is kind of like the bulk of the study, we're trying to design new AV vectors with type 1 myofiber specificity. So you can see here some vectors were carrying the GFP and then some TNNT1 gene, a muscle-specific promoter, MHCK7 here and here, and then different microRNA cassettes, so MIR-T1, T2, and T3. So we did that, and then we wanted to characterize type 1 myofiber specificity of these new AV vectors. So we injected, this time via tail vein, 6-week-old C57 mice, so again wild-type mice, with this vector, basically the GFP AAV8 vector, with the different microRNA cassettes. We are exactly at this point. So next week, we're going to harvest tissues for molecular and histological studies. And then the next step would be to test the efficacy and safety of AAV vectors carrying TNNT1 in knockout mice. So we're building the colony now, and 6-week-old knockout mice, we receive by tail vein injection again, the AAV vector carrying the TNNT1 gene. And again, tissues will be harvested 4 weeks after the injection and to assess for therapeutic efficacy, duration of gene expression, and safety. Finally, I would like to thank the members of Miguel Sanesteves' lab, of course, Miguel, who is my mentor, Kevin Strauss, who's our collaborator from the Clinic for Special Children in Pennsylvania, and then all the other people from my lab. Some of these pictures were taken during COVID, unfortunately. And if there are any questions, I'm here. Thank you.
Right. Thanks. So we found some applause sound effects to make it feel more realistic. So that was a great talk, Eleanor. Thank you for that, and you explained those points really in a way that made it easy for me to understand as a non-geneticist. So thank you for that. Are there any questions for Eleonora? Nothing coming through yet. How many patients have this this condition? Humans, I mean.
Not many, but that's why we collaborate with the Clinic for Special Children in Pennsylvania, because they work with Amish and Mennonite communities, and so they have a lot of patients. They're doing a natural history study, and now they have 4 patients, specifically this type of Nemaline myopathy, so troponin. So that's how we collaborate with them.
I see. It's almost a sort of bespoke treatment for this group of patients that you have there locally.
Sorry, can you say that again?
It's almost a bespoke treatment for these patients that you have there in this local community.
Right, right.
any other questions? I'm more than happy for people to unmute themselves if they want to join the discussion live. But otherwise, I think others. Francesco Montoni, do you have any sense what age the therapy is required before thoracic deformity?
It depends, because unfortunately I've seen differences in patients. I saw a kid who was 2 months old and already showed this deformity, while another kid with the same mutation is now 1 year old and it still doesn't exhibit this whole deformity. So even within the same, the same genetic mutation, there are differences. So the earlier the better, of course.
Good stuff. Well, thank you very much. If there are no further questions, then we shall move on to the next speaker. So thank you very much, Eleonora.
Thank you.
So I think up next we have Konstantin Pharmakidis from University of Kansas. And we've got a bit of a sort of the C word coming up for the next few presentations, COVID-19. But I think this is all relevant to our practice at the moment. So follow-up care in myasthenia gravis during COVID-19, comparison of telemedicine and in-person encounters. So this should be pretty useful for us.
Great, thank you so much. Is my screen effectively shared?
It is, yeah.
Okay, great. Thank you so much for the invitation to speak to the Muscle Study Group meeting. It's a pleasure to be able to join you from Kansas City this morning. As mentioned, the topic of the presentation is basically follow-up care in myasthenia gravis during COVID-19, and specifically a comparison of telemedicine and in-person encounters. Let me start with a little bit of background. Of course, the pandemic brought a surge in demand for telehealth services, also investment in technology infrastructure in many medical institutions, and also regulatory changes that made and supported these telehealth visits. So far in neuromuscular disorders, there's more limited experience with telehealth outside of ALS. Myasthenia gravis is of course a chronic disorder and is sometimes provided in distant tertiary care centers. And there may be potential for providing effective, primarily follow-up care for MG patients beyond this pandemic via telehealth. So objectives, so basically, in the study, the objectives were to compare in-person, telephone, and video conferencing, or I'll call them Zoom visits, for likelihood to have immunosuppressant therapy increases or decreases. So, in terms of comparing the care, I think one of the most sort of streamlined and focused ways was to look at one of the things that we all pay attention to when we see these patients. So specifically focusing on the likelihood of changing immunosuppressant therapy between these different visit types. Also look to assess if, within a large university-based neuromuscular practice, the physicians had specific preferences in visit types, to track the frequency of encounter types since March of 2020, and also to look a little bit at the epidemiologic background in Johnson County, which is where we are. Also to analyze age, distance, and MG-ADL score as potential determinants for visit type and visit selection by patients and providers together, and also record any myasthenic exacerbations during this period, and evaluate for any relation to encounter type. So the methods, this was a single center retrospective study of all MG follow-up encounters, in-person, telephone, and Zoom, conducted between March 16th, which is when we sort of went into this new phase, and August 15th, 2020 at our own tertiary neuromuscular clinic. We had, in this period, we had 307 distinct MG follow-up visits, all the types together. And again, I think the number one question that we had was whether visit type influenced how likely you were to have an increase or initiation of immunosuppressant therapy or a decrease or a stop, a discontinuation of immunosuppressant therapy. So there were 225 visits of patients that were on immunosuppressants, and 181 of those visits, there was no change. So in-person visits, all of the in-person myasthenia follow-up visits, in the in-person visits, 81% had no change. In the telephone visits, it was 80%. In the Zoom visits, it was 78%. So very, very similar numbers of no change. Start or increase. In the in-person visits, it was 13%. Start or increase of immunosuppression, 7% for telephone, 12% for Zoom. And then for stopping or decrease, 5, 11, and 8%. And this is a graphical representation of the data. And there was really no statistically significant difference in the change in immunosuppressant across visit type. So another sort of more overall measure was total quantity of clinician documented actions by visit type. And by that, this was sort of retrospective, looking at what was documented in terms of actions taken at the end of the visit. Of course, this is, there's more bias here, but the overall quantity, I'll refer you to the red rectangle here, and you can look at the median in person. The median was 3 actions. In telephone visits, the median was 3 actions. In Zoom visits, the median was also 3 actions. A graphical representation will show that in this orange box and whisker plot right here, which represents in-person visits, there was much more variation in terms of the low number of actions versus the highest number of actions. But when comparing the visit types, there was no clear statistic, there was no statistically significant difference in the number of overall actions taken. When we looked at physician breakdown of visit types, because one of the questions was, do some neuromuscular physicians do more telehealth than others? And this is only for, again, MG follow-up visits for the pre-specified time. So, provider A had 50, B had 54, provider C had 95 visits in the time that we looked at. Yellow here is in-person visits, the white portion of the bar is telephone, and then the blue is Zoom. And again, there was no statistically significant difference in the distribution of visit types, and there was no clear physician predilection. So this is the visit type by month during the pandemic. What you'll see is that, early on, the in-person encounters shown here in yellow, they basically stopped in April. But then by June, July, and August, they had become, again, by far the number one, the most common way of delivering myasthenia follow-up visits. And then sort of the inverse happened with Zoom and telephone visits, where they went up in May, in April and May very, very quickly, but then they have really come down in the last, they came down towards the end of the summer. So this is a graph of the total number of COVID cases in Johnson County over the sort of the same period of time. And what's interesting here is that in July, again, this total number of cases that's in July and in the summer, the slope of the increase in cases has increased. And that's when, the trends in terms of the clinical visits changed. And I think there's a number, of reasons for that. I think there was more knowledge about ways to successfully mitigate the risk of COVID-19. There was also very specific practices in the clinic about not having people wait inside the building, people coming specifically into the visit room and not being, exposed to other people, no face-to-face checkout, and of course, face masks and distancing during the visit as much as possible. And also other factors may have been patient preference, patient fatigue, and wanting to come in. And that's what we saw there. Okay, so age by visit type. So again, to see whether there was any relationship with age and selection of visit type, I will refer you to this red rectangular box. And for in-person visits, the median was 69. For telephone, it was 74. For Zoom, it was 67. This was statistically significant. This is a graphical representation of the data. The possible inference from this is that older patients, the median of 74 had a preference for telephone, even over Zoom or in person, but that's sort of a suggestion. Distance from home by visit type, which is a particularly important measure for us, because being in Kansas City, we have a large catchment area of patients coming from central Kansas, sometimes western Kansas, parts of Missouri, Arkansas, sometimes Oklahoma. So again, I'll refer you to the red rectangle. The median distance in miles was 20 for in-person visits. It was 20 miles for telephone, but for Zoom, it went up to 62. This was statistically significant, and again, suggested that patients coming from further away had a preference for Zoom, something that could continue after the pandemic. Again, this is a graphical representation of the data. You can see some outliers in the dots that are above the box and whisker plots. Finally, the MG-ADL score, and whether the MG-ADL score had any relationship with visit type selection. Again, I'll look at the median data point and look at the red rectangle. So in-person visits, the MG-ADL score median was 2. For telephone, it was 1. For Zoom, it was 2. And this was not statistically significant. This is, again, a graphical representation. So conclusions. So myasthenia gravis telehealth follow-up encounters are routinely feasible. Both changes in immunosuppressive therapy and overall quantity of medical decision-making, these both appear similar across visit types. After dramatic increases in telehealth visits in April and May, by mid-August, telehealth visits were few, and that probably suggests confidence in mitigation strategies, possibly necessity, patient preference, patient fatigue. The MG-ADL score was not different between visit types, although patient age and patient and distance from the clinic were different. The MG-ADL score can be routinely incorporated in telephone, Zoom, and in-person visits. And no MG exacerbations were observed in the sample, so we really can't make a comment about any association between telehealth visits and sort of what might appear to be poor outcome. This study what it does not capture is the patient perspective on comparative benefits and drawbacks of these different visits, patient satisfaction with different visits, and the role of barriers of inadequate computing equipment, slow or no internet access, and of course, lack of technical experience. Specifically with the older subset of myasthenia patients, these barriers with technology could be particularly significant. And finally, more information on patient satisfaction and telehealth outcomes will be needed to shape the regulatory environment that follows the COVID-19 pandemic. And I want to thank my team and my collaborators in this project, and particularly Suzanne Hunt, who is our statistician and was extremely helpful in the analysis of this data.
Thank you very much, Konstantin. Great talk. You've provoked some debate, so I think there's 3 or 4 questions coming in. I think Dan Basshoff had his hand up first. Dan, do you want to? Oh, maybe he put his hand down now. He disappeared. Dan, do you want to talk? Maybe not. I'll read one of the questions from the Q&A then. So from Gil Wolf, Konstantin, I would think the higher number of actions taken with some of the in-person visits does relate to a more complex follow-up issue that required them to come in. This despite not seeing a difference on MG-ADL. So I think you say you'd assume the people coming to see you, may have some reason to come and see you face to face, do you think?
Right, right. I mean, there was more of a range in these visits. Some people came in and they had 6 or 7 sort of actions taken. Other people had very few, but the difference was not statistically significant. There, of course, there can be some selection bias. People come in when they have more going on, and that's absolutely possible. And the other thing is that different providers record actions taken differently. That's why I think that the changes in immunosuppressant is truly like a very objective way of looking at what we're doing with these patients, because it sort of cuts out some of the things that some people may record and others may not. So.
And then we got Michael Hayher. 2 questions. Number 1, did you look at rate of other metrics like the QOL 15?
No, I did not. And in our in our practice, we use the MG-ADL routinely, as you saw, and we do not use other measures routinely in our practice.
And on that point, same questioner, Michael, you noticed 20% of patients didn't complete the ADL. Any particular reason for that? Who administers the metric in clinic?
I think that's provider.
Nice talk, Konstantin.
Thank you. I think that was provider preference. I mean, there's no mandated pathway, but there's provider preference, and sometimes also I think some visits it may be felt to not be necessary, so maybe it's not done. But I think it's a combination of that and provider preference.
Sure. And then the final question here is it's from someone called Anonymous Attendee. Did management decisions change once patient visits? Did management decisions change once patients who were seen via telemedicine later presented for in-person visits?
Yes, so that's I can see that. I mean, there could be a rebound sort of phenomenon by people sort of having a quicker visit via telephone or via in-person, and then you sort of postpone things, and then there's a rebound. That's a good question. That's not something that we looked at, but it is something that we can look at. So that's a great point. Thank you for bringing it up.
I mean, that's more or less what we've been doing in Manchester, almost having a telephone triage for everyone, and then deciding who comes face to face. And one thing I've been doing is seeing patients perhaps a bit more frequently on the phone than I would have done face to face. I don't know if you quantified the frequency of visits and the face to face system versus the telemedicine system.
We haven't, and I mean, I can see how patients that are seen via telephone visits or telehealth visits, there may be like a feel like it might be better to see them more frequently. I can see that. It might provide some reassurance, but that's not something that we looked at.
Fantastic. Thank you very much, Konstantin. And so we're just on time, so we'll move on to the next speaker. This is SAIC2, post-COVID GBS syndrome mimicking myositis. Sai, are you? Oh, I can see you. Great.
Hi, good afternoon, everyone. Thank you for having me. I'm the neurology resident from SUNY Downstate Medical Center. Let me screen my share.
Just a reminder whilst you're setting that up, if people are inclined to do Twittering and stuff like that, if you want to do that and use the MSG 2020 hashtag. And as I say, do feel free to put your hand up at the end of the talks and ask a question face to face. I guess that's one thing missing from the virtual meetings is just that sort of interaction, but we'll try and do our best to facilitate discussion. So over to you, Sai.
Sure. Today I'm going to talk about a case on post-COVID Guillain-Barré syndrome mimicking myositis. So as a disclosure, there was no financial and conflict of interest regarding to this presentation. So at a glance, cases of Guillain-Barré syndrome has been reported in patients infected with SARS-CoV-19. However, all cases occur during the acute phase of infection, that is para-infectious state. So we describe a Guillain-Barré syndrome GBS case occurring in a patient with 3.5 weeks after his first COVID symptoms, from which he had fully recovered weeks earlier. with negative SARS-CoV RNA test and positive antibody, IgG antibodies on SARS-CoV. This case was atypical as the patient complained of severe bilateral thigh pain as a presentation and tenderness preceding his weakness. Also, unlike most robotic cases, we utilize plasma exchange as a treatment to avoid the thrombotic complications, which is common in COVID-19 infection. So let's go into the case. This is a 61-year-old man with hypertrophic heart disease, dyslipidemia, recently recovered from COVID-19 infection. He presented with progressive muscle pain and weakness in all four extremities, leading to inability to ambulate over the past 5 days. Initially, he presented with pain in his bilateral quadriceps and deltoid muscles. Over the next few days, the muscle pain improved except in the quadriceps, but the leg weakness worsened and became more noticeable after prolonged standing or rising from seated position. After that, he had multiple falls and he started to use cane. Examination-wise is significant for proximal muscle weakness, which I will further elaborate it. There's mild tenderness in the right quadriceps to palpation at that time, and reflexes are diminished throughout. Otherwise, there's no monumentation in intact cranial nerves, including extraocular muscle and facial strength. A few days after, he developed mild loss of vibration and proprioception and light touch in distal legs. So this is a strength testing on admission. So on upper extremities, markedly decrease in strength, 2 out of 5 bilateral shoulder abduction, and elbow flexion asymmetric with right 2 out of 5 and left is 3 out of 5. Elbow extensions 2 out of 5 bilaterally. Wrist flexion and extension are 3 out of 5. Finger flexion and intrins are stronger with 4+ to 4. In lower extremity, proximal hip flexions weakest 2 out of 5. Hip adduction and abduction are 4 to 4+. Knee flexions 3 out of 5. Knee extension 2 out of 5. But ankle dorsiflexion and plantar flexions are full strength. On laboratory testing, CK was mildly elevated at 276, and which later normalized in 3 days. CRP was elevated at 80, and also ferritin was elevated at 709. TSH was normal. We sent for myositis panel, which all came back negative. ANA, double-stranded DNA, and SSA, SSB antibodies are all negative. And the GM1 antibody was sent, which also undetectable. We repeat SARS-CoV PCR testing, which came back negative, but SARS-CoV IgG, serum IgG was positive at that time. We proceed with CSF analysis, which showed WBC of 1, RBC of 24, protein mildly elevated at 49, glucose normal 65. Meningoencephalitis panel were negative. We sent for CSF PCR for SARS-CoV, which came back negative. CSF myelopathy panel, which all came back negative at that time. So we proceed with further imaging. The brain imaging show like only mild microvascular ischemia, otherwise unremarkable. Spine imaging, including cervical, thoracic, and lumbar spine, doesn't show any nerve root enhancement or abnormality. We proceed with MRI thigh because of severe tenderness on that muscle, which was unremarkable except from some lipoma in distal vertex intermedius. We proceed with EMG, nerve contraction study at that time. So it show severe primary demyelinating neuropathy in arms and legs with like prolonged distal latency connection block in right radial motor nerve and mild temporal dispersion in median nerve and absence of F wave. But there was no active denervation in EMG. So we show nerve conduction block in right radial motor with a connection velocity of 36. And there's temporal dispersion in right median motor nerve. And also there's absence of F wave in right median nerve. So during the hospital course, the overall clinical picture and workup support diagnosis of peripheral neuropathy rather than myositis eventually. And he got recent transthoracic echo, which show hypertrophic left ventricle with apical aneurysm, and there's increased risk of cardiac thrombus. So as a treatment-wise, IVIG was not considered at that time. And the plasma exchange was started emergently, and his symptoms markedly improved after second session of plaques. At that time, he could use his arms and legs occurrence resistant. After completing a 5 section of plaques, patient almost regained his full strength, and he started to ambulate with minimal assistance. Later, he was discharged to acute rehab center. So as a conclusion, Guillain-Barré syndrome can occur both in acute and post-COVID infectious phase, sometimes with unusual presentation like in this case. So that's why neurologists should be alert to this complication occurring even weeks after the recovery from COVID-19 infections. So lastly, I would like to thank my attending, Dr. Ansiska, for supervising me, and my colleagues, Dr. Chanchi, for participating in this case. Thanks, everyone. That's all for my presentation.
Oh, they must have run out of the applause, but I'm giving you a virtual applause now, even if you can't hear it. Great, so thank you for that. Just remind me. So, what were the key features that mimicked myositis? That I may have missed it.
So he came with like severe muscle pain, cordyceps, and diarrhea before he has weakness.
Right, the pain is what made you anxious about myositis. I see. Anybody have any questions for Sai? Nothing coming through at the moment. I mean, how sure are you that this was related to COVID-19 and not just some sort of false positive or coincidence?
So the presentation wise, it was atypical, but electrodiagnostically it's very consistent with demyelinating disease. So it's better and consistent with Guillain-Barré syndrome. But the MRI study was atypical without nerve root enhancement. There was not significant albuminous dissociation, but electrodiagnostically this is consistent with GPS.
No, sure. What I'm asking is, how sure are you that it was related to COVID-19 and not just some kind of coincidence?
So we have some robotic cases with that COVID-19 infections coming with this GPS pattern, but this is like more 3.5 weeks. Usually they come with most cases, like in reported cases, case series, they come with like about 3 or 2.5 weeks of that. Yeah. Also, he doesn't have any other viral infection apart from these COVID-19 infections.
Sure. Okay. Well, I don't have. Oh, spoke too soon. It's the anonymous attendee again. Perhaps a controversial question coming. I'm not sure. Is it possible the patient had both autoimmune polyradicular neuropathy and a mild autoimmune myositis, possibly the same underlying post-infectious autoimmune pathology?
I would say it is possible, but in that setting, patient has like mildly elevated CK with muscle tenderness. So we could say like concomitant myositis and autoimmune pathology at this time.
I'm always slightly awkward person to convince, but I guess the evidence he actually had myositis is quite limited. Would you agree with that, Si?
Yes, the MRI study doesn't show any abnormality that is that might show the myositis pattern in the contrast study. So I would say it's less likely. It's always possible, but it's less likely in this case.
Great. Well, thank you very much, Si, and thank you for all the speakers during that session. I think we have a break now, if I'm reading the program correctly, until 1/4 to whatever hour you might be on. So take some time to get a drink.
Yes, before everyone steps away, we are going on break, yes, but we wanted to take a group photo with all the panelists here. So anyone here who is a panelist, please turn your cameras on, and we'll take a couple of quick snapshots.
Oh, fantastic.
Yeah. So as that's going on, I'll kind of wait a moment for people to turn their cameras on. Professor Hanna, please do.
Stanley, you of the group pictures, so we'll do the best we can.
Yeah.
We need your picture too, producer Gil.
Oh, trust me, it'll be there.
All right.
All right, so I'll wait a couple more moments for Carolina, maybe Shafiq if he's here. Mazen, Enza. Okay, that's pretty much.
How many can you get on a screen?
25.
Do we need 2 screens of 25?
I may get a second wave, but all right. So let's take a photo in 3, 2, 1. All right, grabbing that now. Fantastic. Okay, we got it. And if anyone at home is, please send your photos in to Liz, how you're watching the conference. You know, maybe you have your cat watching along with you.
Yeah, that would be good. And that would be really good. Yeah.
Great. Make for a good slide. Yep, that's you perfect. Got it. All right, so yeah, we are going on break now. We'll be back in 4 minutes.
Great. Well, I think that wraps things up. And so, yeah, on behalf of myself and Lindsay, thank you very much for all of the speakers during the session. Some really informative talks and some very relevant talks to today's remote practice and related to COVID-19 as well. So thank you very much to everyone and to everyone for interacting and asking questions. So I don't know if
I'm still here, Michael. Did you ever get out of your car?
Yeah, I'm here.
Okay. Good.
I'm here now. I'm sat with my 90-year-old mother.
Oh, well, that's
yeah. She's been listening to the She's been listening to the lectures.
Are you up north, Mike?
I'm in Leeds. Yeah.
Yeah.
Oh,
fantastic.
So yeah, we did a neuromuscular course a couple weeks ago, and I had my 87-year-old mother there in the room with me. She'd never been to a lecture before.
Yeah, was she impressed?
She really was. That shows how you can really fool your mom.
Yeah. Yeah, she's listening to you, Rick.
Yeah. Sorry. Well, I got her. I had her on the screen too. Put her on, Michael. I had my mom on, and she said hi to you.
I've got the camera doesn't seem to be working for me. Gil?
We see you.
Yeah, we're looking.
Mom, can you wave? Mom, wave at the camera.
Hi, Mrs. Hannah.
Hi.
Hi. Great.
Yeah, these guys are all in America. Missouri. She'd never heard of it.
Yeah, I know. That's a tough concept. That's a tough concept. All right. Well,
the Zoom link for the chatting session is in the chat.
Oh, okay.
Session. Because when we close this out, you lose the link. So everybody copy it. And
so everyone goes, are we all going into the cigar room or some other room?
You can. There's some other rooms. There's some flash presenters.
No, I didn't mean I was forcing people. I thought everyone got put in some generic room.
Everybody's going to be put into the sponsor rooms, and then they can go.
And then you can go wherever you want. Right.
And there is a cigar room.
Well, in the cigar room, we may show a video of me at a baseball game. We'll see how that goes. But all right, well, this has been great.
Been really great.
And Michael, I guess we'll just do this now and then meet again tomorrow morning.
Yeah, okay.
I have to go to an American college football game tonight.
Oh, wow.
Yeah.
Don't get injured.
I'm just hope I don't get COVID.
I know, you better have your mask.
All right. Okay.
See everybody in the mingling rooms.
And right. It does take a couple minutes for me to set it up, so just hang tight.
Oh, we're not yet. Okay.
Well, no, yeah. I mean, you guys can pile into the waiting room, but just I'll set.
This is Rick Barron in America. He's a very famous neurologist. Yeah, he's in charge of a whole university that you never heard of.
You don't know why you showed me in my picture.
She's not impressed.